welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session.
Welcome, everyone, to our Advancing Generic Drug Development Workshop, Translating Science to Approval 2024. This is our 7th year of having the privilege to collaborate with CDER's Office of Generic Drugs and its Office of Research and Standards to produce this workshop. So good day, good afternoon, good night, depending on where you may be across the globe. We certainly appreciate your commitment in attending this event from wherever you may be and for overcoming any scheduling challenges. Please note the on-site, it's the slide. Please note the on-site Wi-Fi login information here in the room, and it will also be available at the registration desk. My name is Brenda Stoddard, and I'm with the Small Business and Industry Assistance Program, SBIA, and you'll hear SBIA throughout the day, which operates within the Division of Drug Information, DDI, which in turn resides in the FDA Center for Drug Evaluation and Research. And here's a little diagram of our organization structure. It is my extreme pleasure, along with my CEDA colleagues, to be with you here today. And added to the excitement is the fact that this is our first hybrid AGDD post-COVID. So thank you all for those who are showing up here in person, and thank you also for being online. This particular event, well, all of our events, features an unsurpassed cast of speakers from across the Office of Generic Drugs, the Office of Pharmaceutical Quality, and the Office of Translational Sciences. In addition to the lectures, for those of you in-house, we have poster sessions just outside these doors there by the coolers, where you can engage with the subject matter experts at the close of the day's presentations, and also tomorrow during lunchtime. We have an extended lunchtime period for that. We entreat you to make most of this experience, to attend all the sessions that interest you, learn all that you can learn, ask your questions during the available Q&A sessions, and for online attendees, submit your questions using the on your screen link and QR code. We are also offering live YouTube streaming, so go ahead, invite your colleagues if they couldn't make it. And however, please note that only those attendees who register and attend via the online login will be able to submit questions to our speakers. We are extremely pleased to announce that this two-day workshop offers a total of 10.5 credits, contact hours to physicians, pharmacists, and nurses. The CEs are available to all real-time attendees who attend via the online logins. And I'm emphasizing real-time and online logins because no CEs will be available if you attend via YouTube or if you look at it after the event for post-event viewing. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. CE faculty or speakers are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations to the information that is presented. We have a spectacular lineup of speakers today, like I said, and the planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to their attendance and complete a final activity evaluation via the CE portal, which is ceportal.fda.gov. And for multi-day activities such as this one, participants must attest to their attendance and complete a faculty evaluation for each day. Final activity evaluations must be completed within 2 weeks after the activity, no exceptions. So in this case, deadline for this two-day workshop is October 9th. And we apologize in advance, but are unable to make exceptions to that deadline. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in the loss of credit for this activity. Your NABP profile number should be the 6 or 7-digit profile number assigned by the CPE monitor, and your birth date should be in the month, date format, example 0924. Do not provide your pharmacist license number. Please click the My Account tab and then navigate to Edit Contact Information to verify that your information is correct. Physicians and nurses may then view or print their statement of credit. Pharmacists should log into the CPE monitor 6 weeks after the last session of the activity to obtain the CE credit. Please allow sufficient time. They're not going to have the credit there 24 hours after you submit. And I strongly encourage you to complete the evaluation or evaluations upon receiving our thank you email. So we won't, as we won't be able to assist if you miss the deadline. And again, the deadline date is October 9th to 2024. I have to emphasize the year as well, because sometimes we get requests a year after the event. Okay, so here are the general learning objectives for this workshop, and I'll give you a few seconds to read through the bullets. As of this morning, we have over 2,800 registrants representing 94 countries. Over 42% of these registrants have self-identified as small businesses, and of those, over 30% report having just 1 to 9 employees. As you can imagine, these stats continue to be very important to us at the program because they testify that we are indeed reaching the small business community, and they also validate our mission and the very program itself. And what you're seeing here on the screen is a snapshot of our geomap for today's event. And this map itself is also quite interesting and exciting, for it gives you a really nice visual as to the impact and extent of our outreach. All right, guys, let's do some wake-up work this morning. This is a poll question. I'm going to run through it. Folks online, please do the poll online. And folks in house, show of hand. The question is, how many SBI and FDA employees are not eligible to put up hands? How many SBA conferences have you attended? A, first one. 2, very good. B, 2 to 5. 2, 3, 4, 5. Okay, we're getting there. 6 to 10. Very good. More than 10. All right, we have some stalwarts in the room. And most since 2010. All right, you're eligible. You're not FDA, but you're always with us. No one more than 10 in the audience. All right. I had a prize for you, but you won't get it, All right. For online, we have, well, you guys can see it. 35% says it's their first one. So online people, if you can come to the next one in person, that would be fantastic. So 24% my first one, and 3% all since 2010. Thank you everyone for participating. I thought that was fun. All right. Let's see if this works, Jeff. All right, so let's just take a couple minutes to highlight our upcoming events. October 9th, we have our ICH M12 Drug-Drug Interaction Studies Final Guidance. This guidance is the first globally harmonized regulatory guidance on assessments of pharmacokinetic drug interactions mediated by metabolic enzymes and drug transporters. October 6th, we'll talk about global IDMP, and we'll present an overview of the end-to-end testing results for the Global Pharmaceutical Product Identification Service Operating Model, which generates global PIDs for marketed medicinal products. Then on November 7th, we'll be talking about the REMS logic model in REMS design, implementation, and evaluation. And this webinar aims to inform stakeholders, particularly those who design, implement, and assess risk evaluation and mitigation strategies. So we have many more events coming up for 2024, just 3 more months in the year, but we do have many more. So please go to our website here, fda.gov/CEDAR-SBIA, sign up to our listserv if you're not already there, and you'll be kept abreast with everything that's coming up. Also, if you go to fda.gov/CEDAR-Learn, you can link to SBIA tutorials along with other educational resources, many of which also offer healthcare professional CEs. I am stuck. Okay. This slide provides the QR codes for social media avenues for SBIA social media. Of course, you can scan them on your phones and be taken to the appropriate platforms, and they will also be advertised across the banner during the breaks. So let's turn our attention to some logistics, and I'm going to just fill this out so it's easier for me, to some logistics as it applies to all our attendees. So here is the link for a one-stop shop for answers to your technical questions, downloading the slides, doing the evaluations, and so much more. For online attendees, this is where you came in this morning, so please keep this link. You're going to need it throughout the 2 days. And if you have any technical questions about this conference, please check there first, and you will most likely find the solution. On our one-stop shop page, there are session evaluation links and also the final evaluation survey, which will be activated this afternoon and will be sent in the daily post event. Upon completion of this survey, an attendee can then download a certificate of attendance, which will be used in support of RAPS, SOCRA, ACRP, and SQA credits. Now, the trick is you have to download the certificate once you fill out the survey immediately, otherwise that opportunity is lost. And if you don't even need a certificate, we ask you to please complete a survey because your comments will help guide the design of future events. And again, the survey and the evaluation, everything that needs to be submitted has a deadline date of 2 weeks, October the 9th. All our sessions are being recorded. It will take us about 2 weeks to complete the recordings, but you will soon be able to access them from our website, again, on the website here, on the slide here at the lower left. But in the interim, for your convenience, the YouTube link for both days will be posted on the One Stop Shop webpage at the end of the day. And just to wrap things up and to simplify things, within 24 hours of the conclusion of each day's event, SBIA will send one email to all the attendees for that one day, that individual day. And on that email, you'll have the link for the survey, which is a certificate of attendance, and you'll also have the claim code information for the healthcare professional CEs. And remember, there are 2 different processes. So now for details and logistics for in-house people. Again, this is the Wi-Fi. I'm thinking everybody has it. Nobody seems to be in dire need of a Wi-Fi right now. For Q&A, we have 2 mics, and you get more, more guidance as the Q&A session progresses. 2 mics, so you'll line up with the 2 mics. We'll take turn between the mics and also online there. And as a reminder, please silence your phones. Use the internet sparingly. You're on your own for lunch. We have water if you need water. But you're on your own for lunch. And we do have flyers at the registration desk where, they have, I think, 4 of the local eating establishments and QR codes where you could scan and pre-order your lunch. But the hotel has been kind enough to also arrange a buffet for us downstairs at the cost of $20. And there are flyers in the hallway as well if you're interested in that. It's a little kind of rainy day here, so maybe you might prefer to stay indoors. Please join us for the networking event, which begins after we adjourn for the day. And so we adjourn around 4:35, networking downstairs in the lobby. But please attend the poster sessions prior to the networking session. Poster session starts from 3:35 to 4:35. All right. At this point, I hope I'm not going over too much time, Dr. Caleb. At this point, I would like to acknowledge these individuals instrumental in making this event possible. Many are listed here, but so many more deserve credit. Special thanks to Maria Monroy Osorio and Ufan Wenman, who led the coordination of all the details, to our CE consultation and accreditation team, to our technical experts in the back and our administrative experts out front at the registration desk. But most of all, our heartfelt gratitude to you, the audience. You took your time out to attend this event, and without you, the event, and we're banking on its success, would not be possible. So thank you. And here's a quick drop-in for you guys. We, SBIA, is trying to determine the most effective way of communicating our events to the audience, to the targeted audience. And we need your help, both online and attendees. I'm trying to get at least 100 people to respond to this, more if you can. So please take this survey. Respond from the perspective of when we send events to you, do you read it on your phone or do you do it at your desktop? So take that in mind in your perspective. We have flyers for the in-house people, so you can scan the QR code outside on the SBIA exhibit table. And online attendees, please scan the QR code here, and it will also be advertised during the break. A very special recognition and introduction to my SBIA colleague, Lieutenant Commander Nora Lim, who will be your SBIA co-host over the next 2 days. And now I am ready. We are ready to launch fully into today's agenda. And as a reminder, all our speaker bios are on the one-stop shop. So I am just going to race through bios today so that we can maximize the presentation time and the Q&A time. I am honored to introduce our keynote speaker, Dr. Robert M. Califf, as the Commissioner of Food and Drugs. And as the top official of the FDA, he is committed to strengthening programs and policies that enable the agency to carry out its mission to protect and promote the public health. Previously, Dr. Califf served as Commissioner of Food and Drugs from February 2016 to January 2017. Dr. Califf also served as the FDA's Deputy Commissioner for Medical Products and Tobacco from February 2015 until his first appointment as Commissioner in February 2016. We are truly honored to have Dr. Califf here in person. We know how busy his schedule is, so please let us welcome Dr. Califf.
Thanks so much, Brenda. I know these meetings take a lot of work, and it's much appreciated. If I look a little tired, I've spent the last 4 days chasing my 3- and 4-year-old grandchildren while their parents were taking a vacation at 11,000 feet in Breckenridge, where they live. So just returned last night. It's amazing the energy that 3- and 4-year-old children have. And, so I am delighted to join you to help kick off today's workshop. This marks the 7th year we've held this important event, focusing on the transformative impact on generic development, regulation, and approval as a result of the FDA's generic drug user fee amendments, science and research programs. These meetings take a lot of work and they summarize a lot of information. It's a great chance for me as I prepare for this to summarize from the point of view of the FDA as a whole where we are. And it is important, this is the first year that's included both in-person and virtual attendees after 4 years of virtual-only workshops. That's an exciting development, and I want to thank the Office of Generic Drugs and Office of Pharmaceutical Quality in collaboration with the Office of Small Business and Industry Assistance for facilitating this and once again organizing today's workshop. I also want to recognize the developers and other stakeholders who are participating here today, as well as the industry support of the generic drug development program. The FDA offers many different types of public workshops and meetings, but today's has a particularly important emphasis. At the most basic level, this meeting provides a valuable opportunity for attendees to engage with FDA scientists on topics including guidance development, advanced analytics, and physiologically based pharmacokinetic modeling for generic drug development and equivalence characterization. This is an approach we pursue across the agency in our effort to provide meaningful opportunities for FDA scientists and industry to share knowledge through personal contact and in-depth discussion. What makes this session even more special is the focus captured in the second part of the program title, Translating Science to Approval. These few words highlight an important aspect of the FDA that's often overlooked or misunderstood, and that's the role we play in spurring and supporting innovation. A critical part of our work to advance the health and safety of consumers and patients involves our role in driving scientific innovation that will benefit the public. We're constantly working to develop and adapt regulatory policies in response to and in anticipation of scientific opportunities and changes so they can be harnessed for the improvement of health and medical care. Few areas have as direct a connection to the public in this regard as generic products, both in the personal benefit that generic products provide to patients and the broader advantages they bring through lower costs and increased accessibility as a result of greater competition. I certainly can speak from personal experience to the benefits of various medications I've taken over the years that are available to me in generic form. As a cardiologist, I'm keenly aware of the importance of controlling my blood pressure and lipid levels as I age. This is completely accomplished with inexpensive generic drugs, and I'm sure many of you have had similar experiences. It's no coincidence that this meeting is being held today, September 24th, a date that marks the 40th anniversary of the Hatch-Waxman amendments. That law has had a remarkable impact on the U.S. healthcare system by creating the modern generic drug application pathway and strengthening the generic drug industry, which in turn has provided the public with greater opportunities to acquire less expensive treatments. It's truly a paradigm shift. Consider that when the amendments were enacted in 1984, generic drugs accounted for just 19% of all prescription drug purchases in the United States. Today, generics account for more than 90% of prescriptions filled in the U.S., and as the pipeline from the innovative sector ages, the availability of generics continues to rise. The economic effect is also significant. According to the Association for Accessible Medicines, in 2022, the most recent year for which statistics are available, generic drugs saved the U.S. healthcare system an estimated amount of almost $400 billion. I feel compelled to also point out that a high-quality, low-cost, and reliable generic drug supply is essential to the public health of the more than 7 billion people who live outside the United States. Your success has implications far beyond the needs of the U.S. and other high-income countries. But as in any field, and nowhere more so than in science, changes come fast and frequent, and those changes require continued adaptation. When the Hatch-Waxman amendments created the framework for a generic drug review, most drugs were simple small molecules. These products generally involve straightforward and relatively simple reproducible manufacturing processes. They were easier to characterize and evaluate through traditional methods, including human bioequivalence studies. Today, however, many drugs have more complex formulations, active ingredients, and delivery systems. In other instances, a complex drug may be the one that acts locally on the tissue rather than through the concentration in the blood. What all this can mean is that these products can be more difficult to replicate, develop, and manufacture. And this is where the FDA plays a particularly important role. As the pharmaceutical science advances and the challenges posed by the increasing complexity of formulations and delivery systems grows, the FDA works to provide generic drug developers a clear regulatory process with streamlined, predictable, and science-based information that help reduce the time, cost, and uncertainty in developing these products. FDA scientists work collaboratively with partners in academia, the commercial research infrastructure, and industry to communicate early and develop innovative tools to overcome some of these challenges. The centerpiece of this process, as well as of this meeting, is the 2012 Generic Drug User Fee Act, or GDUFA, and its subsequent reauthorizations. GDUFA introduced critical timelines for application assessment. Most significantly, though, for our discussion here, it established a GDUFA Science and Research Program, which, thanks to the dedicated resources and structured framework for innovation, continues to help propel the industry forward, driving progress in science and technology to enhance generic drug development and approval. The program has consistently fostered early engagement between the FDA and generic drug industry, supported collaboration with generic industry representatives to determine the priority initiatives, facilitated continued engagement with prospective ANDA applicants through pre-ANDA meetings, and provided opportunities for better informed engagement following ANDA submission in meetings between FDA and ANDA applicants to discuss scientific matters. Here I'd like to just say I would encourage all of you to talk this up, because when I'm dealing in the public sphere, very few people actually understand how much work goes into the pre-application system and the importance of the communication that goes on. It's a huge part of the work of the FDA that I think is not well appreciated. At its core, GDUFA research aims to make generic drug development and assessment more efficient while maintaining a high degree of reliability. These efficiencies can make it more feasible for manufacturers to develop generic drugs, which can reduce the risk of drug shortages and facilitates competition that generally reduces the cost of medicines. This enhances patient access to treatment by helping make these products more available, allowing patients in the U.S. to get the medicines they need. Before I return to the evolving landscape of generic drugs, I think it's important to address the occasional criticism and generalization, or perhaps overgeneralization, that we often hear from some who say, quote, generics are just copies, unquote. On one level, of course, that's true. Generics include the same active ingredient formula as a brand name drug. However, the development process involves far more than simply copying ingredients, as you might do in a recipe. It involves intricate scientific work, and by the same token, assessment across various disciplines is required to evaluate generic drug applications to ensure the generic medicines are safe and effective before they can be approved. Both the development and the assessment will serve society best if we collaborate on the principles of the involved science in manufacturing while maintaining appropriate boundaries in the evaluation process. To use a much simpler example, consider what it would be like to replicate your grandmother's famous and probably top secret chocolate chip cookie recipe or the secret formula for Coca-Cola. You couldn't do it by just looking at the cookies or the soda. With a generic drug, however, you're not just trying to replicate taste, you must demonstrate bioequivalence. That's a tough standard involving a complicated process. And it's only grown as the products increasingly involve some of the most advanced pharmaceuticals in the therapeutic armamentarium. They may involve novel delivery systems with products that include precision devices, novel active ingredients such as oligonucleotides, and unique routes of delivery that are relevant to specific diseases or provide more rapid onset of efficacy. Moreover, the process involves more than just scientific review. We also have investigators who conduct facility inspections throughout the world. We're in the middle right now of the largest reorganization in the history of the FDA, largely involving the group that you used to know as ORA that will be known as OII as of October 1st as the reorganization comes into place. And our patent exclusivity team plays an essential role in determining if we can act on applications. Furthermore, disciplined project managers and regulatory project managers help pull all these pieces together. All of this underscores the complexity and enormity of this undertaking as this field of regulatory science continues to evolve. The FDA works to integrate technological advancements and innovative research methodologies to streamline approval processes. Innovative tools enhance the understanding of complex drug interactions and mechanisms, aiding in the development of generics for more accurate, intricate formulations. Robust approaches, including analytical techniques and bioequivalence studies, enable accurate assessment of generic drug quality and equivalence to ensure that generic drug products are safe, effective, and high quality, just like their brand name counterparts. Let me offer one example of a recently approved generic product that illustrates the meaning of FDA-supported scientific innovation and what can come out of years of GDUFA-supported research. In July 2024, FDA approved the first generic for bupivacaine liposome injectable suspension, a locally acting product for pain management. The treatment uses multivesicular liposomes as the drug delivery system. By any definition, it's a complex product. It was the FDA's research to better understand formulation characteristics and drug release mechanisms, which was presented at this workshop in 2018, that enabled the development of a product-specific guidance that lays out clear expectations on an in vivo PK study in healthy subjects, along with comparative characterization studies to enable generic development of this complex product. And last October, the Generic Drug Program launched a pilot that offers new and early meeting opportunities to prospective generic drug applicants who are using model-integrated evidence approaches for bioequivalence establishment in their ANDAs. MIE approaches combine data from in vivo or in vitro studies with scientifically sound mechanistic models to provide more efficient ways of demonstrating bioequivalence in some of the most complex situations facing the generic drug program. I also want to briefly mention how the FDA's research and collaboration extends to other international regulatory authorities in our work to create consistent, scientifically driven international standards. The recent endorsement by the International Council of Harmonization of the first harmonized guidance for generic drugs titled M13A, Bioequivalence for Immediate Release Solid Oral Dosage Forms, is one such example of successful collaboration. This guidance, which represents efforts and harmonization of 22 global organizations, describes the scientific and technical aspects to support bioequivalence assessments for orally administered immediate release drugs. You'll hear updates on all these programs during the workshop, and I hope many of you will take advantage of FDA's many opportunities to engage during the drug development and approval processes. Finally, I want to mention the logistical challenges involving supply chains that we're working to address. For instance, one way we're trying to strengthen our medication supply chain and patient access is by encouraging and facilitating the adoption of advanced manufacturing processes, ensuring that drug manufacturers will have access to advanced manufacturing technologies is vitally important because advanced manufacturing requires a skilled workforce and can help domestic companies operate with lower costs and fewer quality defects in smaller facilities, improving the global competitiveness of U.S. manufacturing. Our Center for Drug Evaluation and Research, or CDER, has developed an emerging technology program known as the Framework for Regulatory Advanced Manufacturing Evaluation Initiative, or FRAME. Its goal is to support implementation of advanced manufacturing technologies and build a robust extramural and intramural advanced manufacturing research program. The program has a purpose to engage with companies that are pursuing innovative approaches to pharmaceutical product design and manufacturing early in the development process. The emerging technology team can help identify and discuss scientific and regulatory challenges prior to a regulatory submission, which can streamline the application submission process. The ETT has facilitated the approval of drugs manufactured using continuous manufacturing for a range of product types, including solid oral dosage forms and biotechnology products. As I hope this brief summary makes clear, there's a lot happening in this area, and we're eager to work with you. If you thought it was simple, just the terms that have been used in this address are really quite remarkable. If we look back 20 years ago, couldn't have been anticipated. This workshop is a key component of our stewardship of the GDUFA program and an example of our dedication to open communications with the generic drug industry and the broader public on this important research. By sharing cutting-edge research, methodologies, and tools, such as the new equivalence approaches, analytical techniques, and PBPK modeling, we seek to advance the development of generic drugs and contribute to the availability and access to safe, effective, and high-quality medicines. Thanks again for your engagement. I know there's a big audience out there who really are more interested in hearing from the scientific experts than the pontification of the commissioner, but I appreciate the chance to summarize a lot of great work done by our team and the great hopes for you all as you make better and better drugs more accessible to the public. Thanks again and have a great meeting.
Thank you very much, Dr. Caleb. It was indeed a pleasure for you to make time on your schedule for us. So thank you so much. Okay, so let's enter into the world of science and complex generic drugs. And what I'll do, I'll introduce each session, all the speakers at once, and then each speaker will come up in turn as they present. So let's start with session 1, our scientific and regulatory considerations for in vitro release test for complex drug products. First, we will have Dr. Abu Mustafa, who is a pharmacologist with the Office of Bioequivalence within the Office of Generic Drugs. Then we'll have Dr. Thilak Mudalij, who is a research chemist from Arkansas Human and Animal Food Laboratory, still within the Office of Regulatory Affairs. And for the third speaker for this session, we'll have Dr. Dongkai Zhu, who is a visiting associate from the Office of Pharmaceutical Quality Research within the Office of Pharmaceutical Quality. So let's first start with Dr. Mustafa.
Hello, good morning, everyone. Thank you, Brenda, for your kind introduction. So I am AGM Mustafa. I am a staff fellow in the Office of Bioequivalence. Welcome to my presentation. In this presentation, I will talk about in vitro release test, IVRT, for in situ gel or depot forming drug products. So what do we want to learn from these presentations? So in this presentation, I will talk about some key challenges in IVRT method development for in situ gel or depot forming drug products. I will provide some key considerations in IVRT method development and validation for this type of drug products. I will also provide an overview of the important submission contents related to IVRT study in ANDA. So let us first understand what are these in situ gel or depot forming drug products. So these are, in simple terms, these are specialized formulations which after administration forms a gel or depot at the administration site, and that drug releases from that depot over an extended period of time. So for this drug product, the in situ form depots dictates the drug release kinetics. So far, FDA has approved 15 new drug applications and 4 abbreviated new drug applications for this type of drug product. You can see that there are relatively fewer approved generics for this type of drug products. There are multiple reasons, including challenges that are associated with bioequivalence study. This slide contains a summary of the bioequivalence study recommendation provided in the product-specific guidances of in situ gel or depot forming drug products. As you can see that for most of these drug products, PSGs recommend in vivo BE studies. However, there are drug products for which PSGs recommend in vitro BE approach also. And in many cases, for in vitro BE approach, IVRT study is a key component. Here we have 4 examples of in situ forming drug products for which PSGs currently recommend IVRT study. Okay, now what is the purpose of IVRT study here? So IVRT study here is the component of in vitro BE approach. The other 2 components are Q1, Q2, qualitative and quantitative sameness, and Q3, which is comparable physicochemical characteristics. In case of in vitro BE approach, the bioequivalence is determined based on the totality of evidence. And here IVRT is supposed to determine or ensure that the rate and extent of drug release, which can be affected by factors such as drug substance, formulations, and manufacturing process, the comparability, make sure that the test and reference products have comparable test and rate and extent of drug release. And by ensuring that, IVRT study mitigate a potentially major bioequivalence failure mode. Now, why is it challenging to develop an adequate IVRT method for this type of drug product? First of all, there is no compendial method available for this type of drug product to follow, and the product-specific guidances do not provide any specific recommendation about the study conditions. Also, there are challenges associated with demonstration of discriminatory ability of the proposed IVRT method. However, for this drug product, the most specific challenge is optimization of the gel or depot inducing conditions. Now, how to address these challenges? So the key to address this challenge is to understand the mechanism of depot formation and drug release for these drug products. The process of depot formation and drug release can vary from product to product. For example, there are some in situ forming drug products that are self-aggregating peptide drugs, and also there are some polymer-based formulations. In the subsequent 2 slides, I will provide illustration of the process of depot formation and drug release for these 2 types of drug products. So in case of self-aggregating peptide drugs, after administration, these peptide drugs are usually administered as solution form, and after administration, the peptide drugs undergo a self-aggregation process. And this self-aggregation process of peptides forms a dense fibrillar network, which acts as a depot. And from this depot, the peptide drug diffuses out into surrounding tissues. So for this type of drug product, the microstructure of this fibrillar network dictates the drug release kinetics. In case of polymer-based formulations, these formulations usually consist of drug substance, polymer, and organic solvent. After administration, the organic solvent diffuses out and the body fluid diffuses in. And this process causes precipitation of the polymer with entrapped drug substance. And the precipitated polymer drug acts as a depot from which the drug releases over an extended period of time by various mechanisms such as initial burst release, which is followed by diffusion facilitated release and degradation facilitated release. And for these drug products, the characteristics of the polymer dictates the release kinetics for this type of drug product. So we have discussed that for in situ gel or depot forming drug products, the in situ form depots dictates the release kinetics. And that's why, unlike the conventional IVRT method, in case of in situ gel or depot forming drug products, the IVRT method requires a gel or depot formation step. To further elaborate on this unique IVRT method parameter, which is gel or depot formation. So in case of conventional IVRT, the drug product samples are loaded as it is onto the IVRT apparatus. However, in case of in situ gel or depot forming drug products, the drug product samples to be used in the IVRT should be in a adequately formed gel or depot state. If IVRT is conducted with samples that are not in gel or depot form or not in adequately formed gel or depot, then the observed release profile will not reflect the true or actual release characteristics of the drug product. Instead, we may observe some artifacts or misleading release profiles. And this will eventually lead to incorrect assumptions on the comparability of the rate and extent of drug release between the test and reference product. So what are the key parameters for IVRT method development for this type of drug product? The parameters are sample preparation, selection of apparatus, sample loading, release medium selections, flow or stirring rate, temperature, sampling time, and specifically for this drug product, gel or depot induction. For each of these IVRT method parameter, one should take a step-by-step systematic approach for selection of the most optimal conditions. And the expectation is there should be adequate justification with supporting data for the selected conditions. And the overall objective of IVRT method development for this drug product is to achieve adequately sustained release profile and at the same time complete release within a reasonable time frame. So what are the key considerations for IVRT method development? What are the key considerations for gel or depot induction for this type of drug product? First of all, one should take an exploratory approach to investigate the factors that can affect the gel or depot formation process. Since it is not practically feasible to exactly mimic the in vivo condition, one should take an exploratory approach, explore various inducing conditions, and including the physiologically relevant conditions. And depending on the drug products, some of these factors are sample amount, the inducing media, sample to media ratio, incubation temperature, and time. And after the trial, after the induction, one should assess the consistency and reproducibility of the gel or depot that are formed using the given conditions. Next, I will talk about the key parameters for IVRT method validation. The two important parameters for IVRT method validation are robustness and discriminatory ability. In case of robustness, we want to see that slight variation in IVRT method conditions do not have significant impact on the results. And there should have pre-specified criteria for robustness. In case of discriminatory ability, we want to see that the proposed IVRT method can differentiate intentionally designed non-bioequivalent experimental test formulations. And during discriminatory ability evaluation, one should consider the critical formulation and manufacturing process attributes that can affect the depot formation and drug release. Now, what are the key IVRT study-related submission contents that we expected to see in an ANDA? The submission contents should cover these key elements: IVRT method development, IVRT method validation, analytical method validation, and pivotal IVRT study. And some typical contents are standard operating procedures or protocols that are effective at the time of a study, the detailed study reports, sufficient experimental details that will enable the assessor to reconstruct how the study was conducted, and all the data and calculations, which will enable us to assess the accuracy of the results. From agency's point of view, all these informations are critical for the assessment process. And in many instances, it is found that the deficiencies are mainly due to incomplete submission, incomplete information in the submission, which eventually delay the approval process. For awareness purposes, here we have summarized some typical IVRT study-related deficiencies that are found in an ANDA for in situ gel or depot-forming drug products. So we have found that in some cases, though gel or depot formation is needed, but the proposed IVRT method, like a depot or gel formation step, and it is not clear from the submission that the samples, the IVRT samples, are inadequately from gel or depot. In some cases, it is found that a random condition is selected for gel or depot induction without conducting thorough exploration of different conditions, including physiologically relevant conditions. And there are some instances we found that adequate justification is supporting data were not provided for the selected conditions, or the justifications were inadequate or incomplete. We have also found examples where the gel formation was inconsistent and irreproducible, which potentially contributed to the observed high variability in the release profiles. Another typical deficiency we found is that in case of discriminatory ability evaluation of the proposed IVRT method, the critical attributes are not considered. So here are some key takeaways from this presentation. First of all, the gel or depot formation is a critical step in IVRT method development for in situ gel or depot forming drug products. It is advisable to conduct a thorough exploration of different inducing conditions, including physiologically relevant conditions. And one should aim to achieve a well-formed, consistent, and reproducible gel or depot to ensure reliable and representative in vitro release profile. For optimization of IVRT method parameters, it should be based on the understanding of the depot formation and drug release mechanism. And during discriminatory ability evaluation of the proposed IVRT method, one should consider the critical attributes which can be related to formulation and manufacturing process that can affect the gel formation and drug release. We have two challenge questions here to assess if we have achieved the learning objective of this presentation. So the first challenge question is, which of the following statements about IVRT So I'll give you some time to go through the options. And the answer is option A and C. So in case of gel or depot forming, understanding the gel or depot formation and drug release mechanism is important for IVRT method development. And in case of in situ gel or depot forming drug products, additional consideration should be given to the gel or depot forming step. Challenge question 2 is, which of the following statements about IVRT for in situ gel or depot forming drug products are false? Select all that apply. And the options are and the answer is option A and E. So in case of IVRT method, it does not necessarily need to exactly mimic the in vivo release profile. And IVRT method validation is distinct from analytical method validation, and it is expected that the submission content should cover both aspects. So I'd like to thank Office of Bioequivalence for giving me this opportunity to talk about this topic. And my special thanks to Hisan, Utpal, and Bing from Office of Bioequivalence who helped me with this presentation and guide me with this presentation. And last but not the least, the assessment teams from Office of Bioequivalence who reviewed these complex drug products. And the basis of this presentation is those assessments. Thank you so much for your attention. Next, I would like to invite Tilok Mudalis for his presentation.
Thank you very much. Today I will talk on nano-size compressed product in vitro release testing. I'm a research chemist from Office of Regulatory Affairs. So I will start my presentation with introduction to drug product containing nanomaterials, liposome and liposome drug product, in vitro release testing of compressed product containing nanomaterials and challenges. Finally, I will discuss innovative method we developed in our lab. So what is compressed product? Drugs that are characterized by complex active ingredient, formulations, or route of delivery. What is drug product containing nanomaterials? Submicron size particles with one or more therapeutic agents that are dispersed, adsorbed, or covalently bound in encapsulated vesicles, capsules, or polymer matrices. Drug-loaded liposomes, polymer nanoparticles, or protein nanoparticles are prime examples for those products. So what is liposome? Liposome is microvesicle composed of lipid bilayer. It has a central aqueous cavity. We can use liposomes to deliver drugs. If active pharmaceutical ingredient is hydrophilic and water soluble, we can load into the central cavity, aqueous cavity. If active pharmaceutical ingredient is hydrophilic, we can load into the lipid membrane. There are many different kinds of liposomes based on the structure of lipid membrane arrangement. They can have a single layer of single lipid bilayer is called unilamellar liposomes. Multiple concentric lipid bilayer is called multilamellar liposomes. And they can have a multiple compartment in single liposomes separated by lipid membrane. It's called multivesicular liposome. When liposome is loaded with active pharmaceutical ingredient, it is called liposome drug product. So how we do in vitro release testing for liposome drug product? So we set the condition for drug release in drug release media. We can control pH, temperature, and media composition and other parameters. Then we add the liposome formulation to the media. So it starts releasing active pharmaceutical ingredient and to the outside of the liposome. Then we collect the sample each time point. Once we collect the sample, we need to separate them immediately. Collecting sample does not mean it stop releasing. Even we taking the sample out, it keep releasing. So we need to separate them immediately and then quantitate the released drug to make a drug release profile as shown in last diagram. But this is challenging process because both nanoparticles and active pharmaceutical ingredients are same size. So it is difficult to separate by conventional technique. And also these liposomes are delicate structure. If you use harsh conditions to separate them, they can break open. So when we develop a drug release testing method, we need to consider all these parameters. There are mainly two different ways to do drug release testing for liposomal formulation. One is as shown in previous slide. We can take sample and then separate externally, either filtration or centrifugation. But both filtration and centrifugation is challenging because active pharmaceutical ingredient can bind to the membrane or liposome can get disintegrated. sometimes difficult to centrifuge because lipids and media has same density, so they don't get sedimented with centrifugation quick enough. Other method is membrane deposition method. We can take a dialysis sac and then load liposome inside the dialysis sac and place dialysis sac in the drug release medium. And drug release out from the liposome released to the surrounding medium, and then we can take samples from surrounding medium. We can do in the container, or we can do this on flow-through system like a USB type 4 dissolution system. But this is also challenging because active pharmaceutical ingredient can bind to the membrane in this case. Also, it's a non-sync condition most of the time. That drug release inside the dialysis cassette that does not come out immediately. There's always concentration gradient. With considering all the above challenges, we set up the condition to develop new drug release testing method. We need to eliminate manual separation, avoid artifact associated with disruption of nanoparticles, and we need to do it automated way and real-time sampling and analysis, because even after we take the sample, active pharmaceutical ingredient keep releasing, we cannot store the samples. So first example I'm showing is application of capillary electrophoresis to do automated drug release testing. Capillary electrophoresis is a very gentle separation technique. It uses buffer-filled glass capillary, and once we inject sample from one side, we can apply electric field and we can separate released drug from liposome-bound drug, and then we can use optical detection like the optical absorbance or fluorescence. Even we can connect to the other detectors and then measure the released drug. So this is automated method, and this buffer we use in this method for separation is compatible with liposomes. And this method need very small volume of sample. It is a fraction of 1 microliter, few hundred picoliters. And also we can do simultaneously take the sample out, separate and quantitate in automated manner. So this is the example of use of capillary electrophoresis for drug release profiling of liposomal doxorubicin. In this case, the doxorubicin is loaded inside the liposome. Once we set the drug release condition, it starts releasing active pharmaceutical ingredient. And then we can set up the instrument to take the sample directly from the drug release vial and then separate from bound drug to pre-drug as two separate peak. And then we can integrate the peak at the end of the experiment and make a drug release profile. Here I am showing a drug release profile of 5 FDA-approved doxorubicin liposomal formulations, and more details available in a recent publication. We can use same method for drug release profiling of visudine. Visudine is a liposomal formulation of vertepropine. Vertepropine is water-insoluble compound. So once we inject this formulation to our blood circulation, it releases as a human serum albumin complex, as it is water insoluble. In this case, we use human serum albumin to assist the active pharmaceutical release as a complex, and we can separate the released drug from liposomal bound drug. So drug release condition for this drug is we need to get 90% of active pharmaceutical ingredient released in 10 minutes. And we changed the vertepropine to human serum albumin ratio and did the experimentation. And even at 1:1 ratio, we can get out 90% of the active pharmaceutical ingredient. In the human blood, we have about 35 gram to 50 gram of human serum albumin, and it has much more human serum albumin than it wanted to release the drug. So this experiment was very successful, and more detail available in recent publication. So next, I'm going to show a different way of doing drug release testing. In this case, we use electroanalysis. So we insert 3 electrode to drug release container or drug release vial, and then we can in situ measure the released drug without taking any sample. So here in this case, we use the redox reaction of doxorubicin, whatever coming outside the liposome. And then redox current is proportional to the concentration of doxorubicin. So in that way, we can measure the drug release in situ without taking any sample, and then we can generate the drug release profile as shown in this figure, and more detail available in our recent publication. We also develop other method. One of them is a chromatographic method. We modify HPLC system to develop a drug release profiling method. Here we use a monolithic silica column coated with polyvinyl pyrrolidine. And this column has 1 micron through pore, so those pores go all the way along the column. And when you inject the liposome to this column, it let liposome pass through the pores and then release active pharmaceutical ingredient, bind to polyvinyl pyrrolidine, and then we can elute them later. So we can separate liposome from released drugs and generate the drug release profile. This is the drug release profile of FDA-approved 5 liposomal formulations. And again, more information available in recent publication. So each of these methods has advantages and limitations. So we have to carefully select the method when we use it. Capillary electrophoresis use very small sample volume, so that's an advantage. And also it's a delicate separation technique because separation happens in aqueous phase. But separation is done by applying electric field, and only charged active pharmaceutical ingredient can be separated by this method. That's the limitation. Electroanalysis is another way to do the in vitro drug release testing. So this is advantage, no separation, no sampling. We can set up the instrument and it measure the released drug automated way. But this method only works with electroactive APIs. So these compounds should have a redox or oxidation reaction. The other method I discussed is a chromatography-based method. This method highly accurate. It takes exact sample volume. If they take 1 microliter, it's exactly take 1 microliter, and it analyzes the sample. Also, we can use the multiple detectors. Some of the active pharmaceutical ingredients are not optical detection cannot be used with some of the APIs. So we can even set up to the mass specs for quantitation. But this method need a specialized column compatible with the formulation. If we use a column which is not compatible, that can break open the liposome and then give erroneous results. Finally, I would like to summarize my presentation. So we developed an innovative method for drug release profiling with a real-time sampling and analysis for liposomal product. Also, active pharmaceutical ingredient specific physical and chemical properties should be considered for selecting in vitro release testing. So we have to look at the, as I shown in these 2 points, we have to look at the compatibility of the liposome formulation with the method. And also detection method has to be compatible with the active pharmaceutical ingredient. And these methods can be utilized for other liposomal formulation with careful selecting the compatible method. Finally, I would like to acknowledge my co-workers at Arkansas Laboratory. They are the one who did all these experiments, including my lab director listed at the end. Also, I would like to thank ORS, OGD, CEDAR. This work done as a collaboration between myself and Dr. Wenley. And also I received technical support and financial support from CEDAR, OGD, ORS. Also, I would like to thank Office of Regulatory Affairs, Office of Regulatory Science. They provide financial support and technical advice for my work. Thank you for listening to my presentation. Next speaker is Dr. Dong Kai Shu. I'm inviting him to give his presentation.
Hi. Good morning, everyone. My name is Dong Kai Shu. I'm a researcher from Office of Pharmaceutical Quality Research, OPQ CEDARS. Today, I'm going to share with you some results about how we develop in vitro release testing for complex drug product. Okay, so everyone deserves like confidence in their next dose of medicine. So pharmaceutical qualities assure the availability, safety, and efficacy of every dose. So this is the overview. So my presentation will have two sections. In the first section, I will talk about why we need better in vitro release testing methods for complex drug products, including challenges and needs. And in the second section, I will talk about our internal approach to solve these problems. In this section, I will use adaptive perfusion as examples, and then followed by a case study and some other potential uses. So like in vitro release testing, also known as IVRT, has been designed for measure the in vitro release rate and extent of the drug release. And IVRT has been widely used for product development, quality control, and bioequivalence studies. So the pictures here are common IVRT methods. So the methods that with blue name tags are compendial methods, like for example, like USB apparatus 127, and the methods with red name tags are the non-compendial methods. So both of them can be used to measure the in vitro release behavior for complex drug product. So the scope of this talk is focused on the challenge of the developing IVRT method for complex drug product like ophthalmic emulsions, protein bonded complex, and also the long-acting injection product and nanomaterials. So you might wondering what a good IVRT method, what kind of IVRT method is a good considered as good. So a good IVRT method needs to be reproducible, which includes being precise and robust, and also it needs to be discriminatory, which including like sensitivities and selectivity. So the goal of IVRT, like I just mentioned, is to measure the in vitro release. The goal of IVRT is to measure the in vitro rate and extent of the drug release by separating the free drug from its other carriers. And the common ways to use this is to membranous diffusion. So which is, and this method can be applied to many type of drug product like Dr. Robinson liposomal injection and paclitaxel protein bonding injectable suspension, nano emulsions, and nano suspensions. So, however, the membranous diffusion does have the limitations because it's governed by the concentration gradient from the high concentration to the low concentration side. And also the diffusion membrane is slow. So usually it takes about 2 to 3 hours for 90% of the drug come across from the membranes. So compared to the drug release, which usually take about within a couple of minutes. So the membrane diffusion method maybe become a really limiting step and cannot capture the entire process. For example, the release process you saw may not be the truly release process. For example, some of the complex formulations that exhibit multi-phase release, like birth release and the earlier stage, the membrane diffusion method may not be captured these features. So now I would like to use the ophthalmic emulsions as examples to further discuss these questions. Ophthalmic emulsions, because it's considered as one of the type of the complex drug product, and it can be challenging because it may contain active ingredients like cyclophorin, and also ophthalmic emulsions have complex dosage form and may exhibit multi-phases release characteristic. And also ophthalmic emulsions, it has contained the complex route of delivery, which is through the eyes. So all those features that make ophthalmic emulsions is a good example for complex drug products. So here is the result that we did for the that we use reverse dialysis as the IVRT method for measure the in vitro release behavior for diphosphorin ophthalmic emulsions. The result on the red shows that in these studies we test like 2 different formulations with different globule size distributions. On the right side, you can see there's no discrimination was observed between this formulation in terms of the release profile. And also there's no multi-physics behavior was observed. And because of this too slow release rate, product degradation was detected. So this kind of highlights the limitations of the traditional dialysis-based IVRT method. So the challenge with the current IVRT method can be divided into two sections, which is reproducible and discriminatories. So from the previous example, we saw the reproducible is well presented in the result by seeing this really tiny error bar in the figures. So how can we improve this IVRTs? So there might be different ways to do that, to tackle this question. And for example, like by developing any other type of in vitro release testing system. So here is an example that I'm going to talk about is that what we did in our lab. This IVRT method named as adaptive perfusion system. So adaptive perfusion system is utilizing the tangential free filtration principle to study the drug release. So compared with the normal filtration process, there will be the feed flow for this method will be parallel to the membrane filters. So there will be really less accumulations on the membrane surface. So anything that accumulates on the surface will be swiped out by the feed flows. Also, anything that's smaller than the pore size of the filter will be forced to migrate through the pore size and to achieve the separations. So there's two vessels are used in this method. One is named as retentive vessel. One is named as permeate vessel. So the retentive vessel is used to study how much drug was remaining in the system, and the permeate vessel is used to study how much drug was released in the system. Because the entire system was recording, so we are easy to calculate the rate and extent of the drug release. So this is a schematic of the adaptive perfusion system. So if you see in the middle, the blue vessel is the retentive vessel, and where you inject samples here and mixing with the dissolution medias. And the gray vessel on the left is a fresh pump reservoir. It will continue to pump fresh media to the retentive vessel to maintain the constant volume in the retentive vessel, which is very critical to calculate the drug removal rate. And the green reservoir on the right is a permeate reservoir, which is to collect the drug release the release drug. And let me explain how this system works. So at the beginning, so you inject sample into the retentive reservoir, which is like the red line here, and the mixture, and then pump through the TFI filters and go back to the retentive reservoir, followed by the blue line. And the back pressure controlling valve here will create a transmembrane pressure that is perpendicular to the feed flow inside the TFI filters. And this pressure will serve as a driven force that forces those free drugs that's smaller than the pore size of the TFI filter to go through the permeate pathway and collect by the permeate vessels. And because of the continuous pumping from the fresh medium, the liquid volume in the retentive reservoir will be stay constant. That is really critical. So for us to calculate the drug release rate. And here is a case studies. So we are using adaptive perfusion as an IVRT method to study the diaphragmatic ophthalmic drug ophthalmic emotions, and we intentionally create 3 formulations that with different globule size distributions. And in this study, we also include the pure drug solution and drug-loaded micelle solutions. And on the left figure, you can see the rate and extent of the drug has been removed from the retentive reservoir. And on the right figure, you can see the in vitro release behavior for these 5 formulations. So what we can see from the left figure is that really like 80% of drug has been released from the solution in less than 10 minutes. And similarly, what we saw from the red figures is that almost 90% of drug has been released to the retented to the permeate vessel within 30 minutes. So both of the figures shows that adaptive perfusion can achieve the fast release, and the figure on the permeate side too shows that adaptive perfusion has a good discrimination capabilities for those formulations. And we also compared adaptive perfusion with traditional dialysis methods with using the pure drug solution and two formulations that have small and large globule size distributions. So as you can see from the left, adaptive perfusion, you can see how fast adaptive perfusion shows drug release compared with the traditional dialysis. And this, the red figure also provides more direct evidence, shows that adaptive perfusion is a fast and discriminatory IVRT method for ophthalmic emotions, which is typically one type of the drug product. So now we are kind of confident that adaptive perfusion is a reproducible method, is an IVRT method that's reproducible and discriminatory. So after that, so we have one step forward, so where we implant the automation function to the adaptive perfusion method. So the figure on the left is our automated adaptive perfusion system, where we design our own programs that can achieve automatically like precondition process experiment and watch the system when the experiment complete. And we also develop an algorithm that can automatically maintain a constant volume in the retentive reservoir, which is, like I said, very critical to calculate the drug removal rate during the experiment. So this automation function dramatically reduces the human burden and allows the result to be consistent even if the system was operated by multiple persons. So the figure on the right shows that this automated adaptive perfusion system can generate similar result, compared with the manual operations. So this shows the result generated by this system is also trustworthy. So in addition to ophthalmic emotions, there's another category of complex drug product, which is formulations that can contain nanoparticles, like paclitaxel protein bonded injection suspensions. So the challenge is for this kind of formulation is that we are lacking the technologies that can sensitively detect the rapid release process of these formulations. And we are exploring the IVRT method can help us understand the role of electrolyte in stabilization and release mechanism. So another category is, in this case, is Dr. Robinson liposonic injections. So the challenge for this kind of formulation is that its delayed release mechanism is difficult to measure using traditional IVRT method. And also, like a lipid nanoparticle is kind of growing platform for the drug deliverers, and we need better IVRT method to understand these formulations. So because of the liposome shares many similarities with the lipid nanoparticles, so Dr. Robinson liposonic injection can be really serve as an ideal platform to study and facilitate the assessment of future lipid nanoparticle submissions. So here is a summary. So IVRT approach for complex drug products can be challenging and needs more concerted effort to improve. And a new IVRT method was developed that can improve understanding of the in vitro release behavior of complex drug products. And lastly, and we really encourage the development of more innovative and fit for purpose for IVRT methods. So here's the acknowledgement. I really want to thank my always mentor, Dr. Xiaoming Xu, for his guidance, and also all the colleagues and collaborators. So, and here is the challenger questions. So true or false? To develop a suitable IVRT method for complex product, compendial apparatus should be used. And the answer is false. So here is the second challenge question. So which is the following is not critical for developing a suitable IVRT for complex drug products? A, sensitivity, B, selectivity, C, robustness, and D, in vitro in vivo correlations. So, and the answer is D. So thank you for your attention. And this is all finish all the presentation. I will invite all the speakers and panelists to the stage, and we can start the QA section.
Thank you very much to our speakers, and we would like to invite our extra panelists to join us. Dr. Hee-Sung Chung, lead pharmacologist with OGD's Office of Bioequivalence, and Dr. Chaomin Zhu, Division Director of the Office of Pharmaceutical Quality Research within the Office of Pharmaceutical Quality. So as a reminder for the in-house, we have 2 mics. Please don't feel shy. Come up to the mics, we'll take turns. Hopefully we have people to take turns. And then we'll go to 1, 2, and then we'll go to an online question in the back and start the whole circle again. So let's start with that, and I will go down to the floor. Not on the floor, but you know what I mean. Anyone in-house? So exciting.
Thank you for the presentation. It's very informative. So I have 2 questions. The first one is for the polymer-based depot long-acting injectable. There are normally like 3 stages, right? Like Dr. Mostofa said, like the degradation, and then the diffusion and the burst release. So does the agency expect like one method or multiple methods to cover different stages of release? Because from our experience, it has been very challenging to have a method to mimic the actual release in vivo.
So the question is, in case of polymer-based formulation, the mechanism of drug release consists of different stages. The initial burst release, which is followed by diffusion facilitated release and degradation facilitated release. So the question is whether we expect that the proposed IVRT method exactly mimic all these release mechanisms. So actually, as I mentioned, that we do not expect that the proposed IVRT method would exactly mimic the in vivo release profile or the release mechanism, but we want to see that the IVRT method, with the proposed IVRT method, the basic characteristics of the drug product, the basic release profile of the drug product is not changing. And there should be, if possible, we want to see that the mechanisms, this initial burst release, and then the more sustained release profile can be replicated. But if with the proposed method, if a sponsor can demonstrate that their proposed method can differentiate the factors which can affect the release profile, then with those justification, that proposed method can be considered acceptable. So we cannot actually dictate what it should be, the method for what it should be, but the applicant has the flexibility to come up with this method, and they have the flexibility to provide justification for the acceptability of their method. But yes, it is definitely advisable to try different conditions and try to see if it can have the relevance. The proposed IVRT method has the relevance with the in vivo mechanism. I hope you have.
Thank you very much for that answer. Anyone else? Normally, if we have a long line, we'll take turns, 1 question each, but it seems that you have the floor. Was somebody raising their hands just now? No hands. You got to come up to the mic, please. Let's go ahead with your second question in the interim.
So for those like long-acting injectables that like release over months, we have developed some like accelerated methods, like short-term method for dissolution. Does the agency also expect like a real-time or long-term method for like a comparison purpose or like a QC or release purpose?
So for QC purpose, definitely the consideration would be different, but for bioequivalence purpose, if it is as a part of the in vitro bioequivalence approach, then definitely, as I mentioned, the expectation is not that the release profile of the IVRT needs to exactly mimic the in vivo profile. As you mentioned, like in some cases, it can be as long as 1 month. So we don't want to, it is not practically feasible. We don't want to see, we don't expect to see that. But we definitely with IVRT method, the release profile would be expedited. But this expedited release profile should have relevance to the in vivo condition. And with expediting, it should not alter the basic release characteristics or the basic release mechanism of the drug product. Then, as I mentioned, that it can give us some artifact release profile, and that could be misleading for the comparability of the rate and extent. And so applicant has the flexibility to come up with their release profile, and under the given condition, we will see whether that release profile is actually adequate enough to potentially differentiate a non-BE test formulations. So it's like case-by-case basis, we have to see the total picture, then we can see, we can assess that whether it is acceptable or not.
Thank you.
Thank you, Dr. Mustafa. And
Okay,
question at mic 2, please.
Okay, my question is regarding generics, like the Me Too products. Usually we look at the USP for procedures for release criteria. In case of a novel drug or something being developed, do you share, as the agency, do you share the typical processes that some of the leading companies are using to a small startup company that's trying to develop a product?
So I can maybe try to answer this question. I think also internally, it'd be great if we have a dissolution database-like system for these complex products, or if we have like a standard method that industry can use. But the reality is that we don't have that. So, you can start with certain methods, right? I think typically people may try USP2 or USP4 if it's, I'd say, emulsion product, for example. But we have also shared today that there's research on different methods that applicants may try using. So we welcome new approaches that you may be considering, and then you can approach us with those methods. But again, we don't have a set method for specific product. That also provides some flexibility for the general industry to come up with different methods rather than sticking to one method, which may or may not work for your product. Thank you.
Thank you, Dr. Chong. Let's go online for a question, please.
Great. We have an online question for Dr. Xiaoming Zhu. Is IVRT demonstration done once only, and does that experiment replace PK study?
That's a very interesting question. So typically we know the IVRT, as you've seen today, we discuss a lot of novel methodologies. And the reason why we need this kind of novel methodologies is because of the challenges that we recognize with the existing IVRT method, as the previous question was alluded to about the USP method. So when we are looking at these challenges, especially if we desire the IVRT method to be used to support, for example, bioequivalence, demonstration of the bioequivalence, then we certainly recognize the difficulty associated with its development that's needed. So for this reason, so typically, at least for now, the IVRT for demonstrating bioequivalence can be considered, for in supporting of the study in the initial kind of filing submission. So certainly there's a kind of the other subsequent question is, can this method also be used for batch release, and then as part of the continuing to release batches? You would think, a good method that suitable for demonstrating bioequivalent would also be suitable for demonstrating quality assurance, QC. But we do recognize the challenge is still there, and we desire at some point to bridge those gaps. But for now, what we are limited discussion is looking at the method to supporting bioequivalence. So for those reasons, those as a one-time study with adequate justifications, with adequate support for validation in order to support that it can serve in lieu of, for example, a clinical endpoint or PK-based BE approach. So that's, I think, the current view on how the IVRT may be used. So hopefully that answered the question.
Thank you, Dr. Su. Mike 1, please.
I have a question for the last speaker. You have two questions. The least critical factor for the RT. So you said it's the in vitro numerical correlation is not critical. So I just wanted your comments. Why is that?
Thanks for the question. So the challenge question is saying that which is not critical for developing IVRT method for when you not critical for when you're developing IVRT method complex drug product. The reason the answer is the in vivo correlation is not that doesn't mean that it's not critical. It's because like if we really using those to require the IVRT method to achieve the IVRT standard, so it will be really hard for us to for the applicant to develop such kind of like IVRT method. So it's not doesn't mean it's not critical. It's because the standard is very high. So we have to, besides to achieve the IVRT, we have to first to think about the sensitivity and selectivity and robustness and process for when we develop the IVRT method for the complex drug product.
Thank you, Dr. Su. Next question.
Thanks. Great talks today. Quick question. Microspheres, PLGA, just curious if you could get an update on any of these technologies for the IVRT are applicable towards that, or if there's anything that you guys did not talk about today during your presentations that might be an ongoing area of research. Thanks.
Yeah, we didn't present on the PLGA microsphere. So, but as you know, last year we had 2 approval of the generic PLGA microsphere product. So part of that, I really attribute to the GDUFA research program that a lot of research has been done at academia laboratory to kind of support. A major part is the how do we measure the release from those long-acting injectable products. So without going into detail, I would refer to you to some of the publication that FDA and collaborators has already published, especially in the microspheres domain. University of Michigan, Purdue and Connecticut, and multiple groups, and UT Austin, they have a lot of research that's in that area that we think can be very helpful for the generic development. So I will refer you to some of those published work already.
Thank you, Dr. Su. So for the benefit of those in-house, we do have several guidances, the cover sheets with the QR codes outside that are relevant to the talks that you're going to hear today and tomorrow. So please take a look at them when you step outside. Also, you're here. Please don't be reticent. This is the opportunity to ask your questions. And don't depend on catching them outside the door, because you may not get that opportunity. So please, this is the forum to ask your questions that will benefit everybody online and in-house. You're here. Don't say, I'm going to wait until tomorrow. Do it now. Online question, please.
We have an online question for Dr. Dongkai Zhu. Are there any ongoing efforts to standardize or validate the adaptive perfusion method across different labs or institutions?
Thanks for your questions. like I described, the adapter, this method we're just also exploring, and we have been testing many formulations like emulsions, and also plan to do some more work related to the protein bonded drug product for injectable suspension, or also another type of formulation. So when you're concerned with like validation process, like so it's still very critical to consider the robustness and to think about the reproducibility and the discriminatory for this such kind of IVRT, this kind of IVRT method. Thank you.
Yes, Dr. Zhu.
Yeah, just to add, so I hear the words in the question, standardization and cross-laboratory. So I think there are different aspects. A standard, when we're talking about standard that require, a significant effort. So for example, USP or ASTM or ISO. So those, I think at a higher level, that means the methodology, equipment, and all the associated, practices are can be replicated in other laboratories. So, and then there's also that the method is reproducible in another laboratory, which is certainly a relatively lower bar to achieve. And I think as what we are presenting today on some of those methodologies is really not to intend to show, this is a method you need to use as if now, but rather as an encouragement for you to consider developing your own methodology, maybe using this as a starting point as you are developing the methodology. And then certainly we cannot anticipate what the product you're going to use, which means what evidence may need to be provided in order to support the approval decision. So those, we think it's your responsibility to take a step further from any of the methods that's described today, or your own method that you have developed in-house, to really show that the method actually can tell the agency what the results is trustworthy, it's reproducible and others. So I think as of now, what we are doing internally, we're continuing the research. We certainly want to make sure the method can be used by others, but it's not something that FDA are necessarily primed to do, of develop, a standards that, we certainly want to share with you, but ultimately to adopt, to use it, it's really up to you to use it. And then again, we really encourage you to develop your own methodology if the existing methodology is not suitable. So that's something, I want to really come to comment on that. Hopefully we can see more novel methodologies available.
Thank you, Dr. Su, for that additional perspective and exhortation. Okay, mic one, please.
So it's a really great talk, and thank you very much. So my questions came from some challenge we have. Let's say we develop product, gel or depot, and use it surgically, not just like topical. And extended release, local release is critical. And in that case, in vivo release, the mechanism, the rates are very unique for the product. I can have two products, they have very different release, but develop in vitro method, they might be the same. It's my question is, what are your thoughts? How can I develop this in vitro method to reflect somehow the in vivo method, in vivo mechanism, so I can tell these two drug products equivalent or not from in vitro study. So your thought about that?
So the question is, I would like to repeat the question first. So the two products that are developed, maybe in vivo they are not equivalent, but by doing the in vitro study, we found that the release profiles are similar. So the challenge is how to tell that they are actually similar, right? Am I right? Okay. So that's why the in vitro BE approach is actually the totality of evidence. So here we have it is a multi-pronged approach. So it is not entirely dependent on the comparability of the rate and extent of drug release, but also there are other components of it, which is in this case, formulation similarity, the Q1, Q2, the qualitative and quantitative sameness of the formulation. And also another important aspect is the physicochemical characteristics. So if you see the PSGs of this drug product, there are multiple physicochemical characteristics that we want to see that they compare between the test and reference product. And with addition to that, we will also want to see that with IVRT, and definitely that IVRT should be adequate enough, and we want to see that it has the discriminatory ability to detect a potentially non-bioequivalent test formulations. So based on all these different approaches, we can mitigate the potentially major bioequivalence failure moods. There are different, there are multiple ways that we can actually make a formulation which is not bioequivalent. And by cross-checking all these different things, we are actually kind of mitigating the risk of approving a non-bioequivalent formulations. So that's why it is a totality of evidence approach. And in addition to IVRT, multiple factors are considered here, multiple things are checking out. And for IVRT method also, since we want to see that under the given conditions, the IVRT method, is it actually really adequate enough to identify any potential non-bioequivalent test formulations? And that's why IVRT method validation is very critical. So in many cases, we found that the deficiencies are related to IVRT method validation. So the IVRT method validation, we found that the critical factors, either formulation related or manufacturing process related factors that can affect this depot formation and drug release were not considered in the IVRT method validation. Since the IVRT method is not validated adequately, we actually usually ask applicant to come back with the supporting evidence that the IVRT method is validated. So that's how actually we want to make sure. But definitely the best, the most confirmatory thing to do would be the in vivo B study. But by giving, and definitely when we are actually giving the options of in vitro B approach in the PSG for any given drug product, we are actually, before doing that, we are trying to make sure that whether by taking this in vitro B approach, is it practically feasible to ensure the bioequivalence of the drug product. So that's why for not all the drug product, we are recommending in vitro B approach.
Thank you very much, Dr. Mustafa. Okay, mic 2, please.
I'd like to add actually.
Oh, I'm sorry.
I'm not sure if your product is actually like same dosage form or intend to be different dosage form, but want to show bioequivalence. Then I think, whether you're going for ENDA pathway or 505(b)(2) pathway, I think there's a difference, right? So if you're thinking about a generic product, then the starting point is like Dr. Mustafa mentioned, you want to start with the Q1, Q2 product. In case of topical product, then we start with the no difference standard, right? If you go back and listen to CRCG presentation by Dr. Sam Rainey from ORS, he gives a very good presentation about how to do the IVRT study, what to consider in developing the method, etc. And so he would then, he goes into explaining you could have different formulations that come back and show same results in IVRT, but that's not what we are looking for in the generic area. And so for generic field, you want to start with the Q1, Q2 formulation for these complex products as applicable. And oftentimes PSG will indicate whether you should start with the Q1, Q2 product, right? And then use that and then develop the IVRT method. And at the same time, ensure that your IVRT method is discriminating the difference in formulation, different strength, different manufacturing processes, so then the method is sensitive to detect the difference, and also then at the same time confirm that test product and the reference product are bioequivalent to each other. Thank you.
Thank you, Dr. Chang. Okay, we have time for one last question from Mike 2, please.
Yeah, thank you for the nice presentation and discussion. So I have a technical question about the same condition to study the release rate from complex formulations using these advanced IVRT methods that you described. Most of the water-soluble drugs, the driving force, mainly concentration gradients from this complex formulation. But for hydrophobic drugs, maybe this formulation is distributed in the body as is, and the release mechanism in the body might be depending on biological factor. So maintaining a same condition during, for example, adaptive perfusion or tangential flow filtration or electrophoresis may have a detrimental or destructive effect of the formulation. So how we can develop a method that is give us like a release rate that can be used for quality purpose. However, it has no relevance to the bioperformance.
Yeah, so it's a great question. I really love the topic about sync versus non-sync conditions. I think it's a great debate. We know typically for solid oral drug product, sync condition is very critical because maintaining the GI, the drug have enough solubility, dissolution rate before it can be absorbed. However, we know for some of the complex product, particularly delivered through very complex route of delivery, such as through topical, through ophthalmic, inhalation, locally acting, and the list can go on, that the limitation of the exactly following the sync conditions may actually impose a constraint to the drug release that may or may not be relevant to the actual condition of use. So I'll give one example. For the ophthalmic, let's say you deliver a suspension product, and the suspension, let's say, is about sub-micron in the nanometer size range. And then you have another formulation that is 5 micron in size. Now, if you run a sync condition experiment of a dissolution using apparatus 2, for example, you will see equivalent release profile in that measurement condition. Now, the question is that results meaningful or useful? If you know the reason you develop a nanoparticle formulation is to enhance the rate of dissolution, because the suspension you apply to the eye only has about less than 2 minutes of time to stay on the eye surface. So ensuring that the drug can dissolve very rapidly in that short duration is more important than to measure the complete release, let's say within 1 hour of a dissolution testing, right? So this example shows that maintaining the same condition, let's say, to ensure you testing 2 products, if you actually get the same results, is that meaningful? So in this case, non-sync condition may actually tells you what is a tendency for the drug to dissolve in a very rapid fashion. Actually, we have published some work related to this that, even a few hundred nanometer differences, you can actually drive the drug release to see the difference. Some can dissolve within 10 seconds, which is very desired, right? And some will release in 5 minutes. So then, relative to the eye condition, the residence time on the eye surface, when you put all of this information into consideration, you need to make a determination which is more important, which is more relevant. So certainly, certain conditions, sync condition allow you to have more reproducible results, but also at the cost may not be relevant or provide useful information. So it's something that really need to be considered during the development. And as part of your submission to the agency, don't be afraid to put in the justification. Don't be afraid to put in justification and reasoning to say, why this is not needed and why this is useful. So it's really case by case. So, I didn't answer really your question, but it's this topic that I think need more discussion, needs more, I think, consensus about, what is really important.
Okay. Thank you very much. Thank you to our panelists. This brings us up to a break. We want to stay on time, so please join us back here at 10:30. And refreshments are in the lobby, posters are out there. in the hall. Thank you. Okay, everyone, thank you. We are a couple minutes later, but that's fine. We'll catch up. Thank you for returning. Some people like to flee and don't come back, so thank you for returning. We're going to start our session 2, Research to Support Guidance Development for Topical Drug Products. Our 3 speakers are going to be Dr. Megan Kelchen, Senior Pharmacologist with the Office of Research and Standards within OGD. Then we'll have Dr. Eleftherios Sakalakou, Lead Pharmacologist from the Office of Research and Standards, and forgive me for not pronouncing it correctly. And then we'll have Dr. Tanaz Ramazanli. I'm trying to talk so fast, forgive me. Senior Pharmacologist with the Office of Research and Standards. So let us begin with welcoming our first speaker, Dr. Megan Kelchen.
Good morning. My name is Megan Kelchen, and I will be starting the session by providing an overview of the current trends and product-specific guidances for topical products applied to the skin. In my presentation today, I'll first describe how research contributes to the development and revision of product-specific guidances for topical products applied to the skin, and then identify some scenarios where it may be beneficial to get feedback from the agency during product development. So let's first talk about some bioequivalence approaches for topical products. The first is comparative clinical endpoint bioequivalence study. This is an in vivo study in which the test product is shown to be as efficacious as the RS, and both products are shown to be as or superior to a placebo control. The next is a characterization-based bioequivalence approach. This is a weight of evidence approach that compares the formulation, microstructure, and performance of the test and RS product. Next is a vasoconstrictor study. This is an in vivo study comparing the skin blanching effect of the test product versus the RS for corticosteroid products. And finally, a waiver of in vivo studies. And a waiver is typically recommended for products that are solution-based where bioavailability is considered to be self-evident. And for the purposes of the discussion today, I want to highlight that these bottom 3 approaches that are shown in the box here are considered to be efficient bioequivalence approaches. So shown here are some bubbles that include the 4 bioequivalence approaches I just discussed. And you can see here that in the overlapping section, that many PSGs for topical products include multiple bioequivalence approaches. So for example, PSGs that recommend a comparative clinical endpoint study often include an efficient bioequivalence approach, such as a characterization-based approach or a waiver of in vivo bioequivalence studies. Now, when we think about how to prioritize PSG development or revision, it's important to note that we first prioritize PSGs for newly approved NDA products. And then we focus on prioritizing PSG revisions for products where efficient bioequivalence approaches are not yet available. So in the context of this slide, the highest priority for PSG revision at this time are PSGs where we only recommend a comparative clinical endpoint study. Now, it's important to note that if a PSG does not include a characterization, or if a PSG only includes a characterization-based bioequivalence approach, that doesn't necessarily mean that an alternative, more efficient approach would not be acceptable to the agency. It may just mean that we have not yet revised that PSG yet. And so we encourage you to discuss any proposed alternative approaches with the agency through a product development meeting request. Let's turn our attention now to characterization-based bioequivalence approaches. I've talked about the components of a characterization-based bioequivalence approach in detail at last year's SBIA presentation. But briefly, at this time in PSGs for topical products, we commonly recommend no significant difference in formulation, comparative Q3 characterization, and an IVRT study. And then depending on the complexity of the dosage form, we may also recommend an IVPT study or systemic PK study. And so focusing in on the no significant difference standard in a bit more detail, it's important to note that the no significant difference standard is a comparison of the test formulation compared to the RS. And it's based on the principles for assessing Q1, Q2 sameness, but it also considers certain differences that have been previously determined to be acceptable based on scientific evidence. And I've given a separate presentation that focuses only on the no significant difference if you want to learn more. So in PSGs for topical products, currently we recommend bioequivalence approaches for for products that meet the criteria for Q3 sameness, which would be a characterization-based bioequivalence approach. And we also recommend bioequivalence approaches for products that meet the Q3 difference criteria, which would be a clinical endpoint study or a vasoconstrictor study. I want to mention that we are currently developing bioequivalence approaches for these Q3 similar products, and those efforts are currently underway. So OGD is constantly investing in research to streamline PSG recommendations over time across similar products. And I'll discuss a few of examples of this in the next slides. But I want to mention that other examples of research will be presented at other meetings this fall, for example, at the AAPS meeting in October. So let's first start with the PSG for topical aqueous gels. This slide is a slide I presented at last year's SBIA presentation that shows the components of a characterization-based bioequivalence approach in PSGs for topical aqueous gels. And so for the purposes of today's discussion, we'll use globule size distribution as a measure for the complexity of the dosage form. And you can see that for the majority of these products, when we do not recommend globule size distribution, we also do not recommend an IVPT or PK study. And the deviation from this trend is Dapsone topical gel. In September of 2023, we included additional recommendations such as IVPT and PK for these products based on our understanding of the site and mechanism of action of this drug product. However, over the last year or so, we've developed a more comprehensive understanding of the structure of the drug product, as well as the impact of this structure on the performance. And Dr. Sacalozzi will talk about how we use modeling and simulation to do this in the next presentation. But for now, I'll just note that based on this research, we were able to revise the PSG in February of 2024, removing the recommendation for IVPT and a PK study from the characterization-based bioequivalence approach. And so now here on the side, you can see the recommendations for topical aqueous gels for a characterization-based approach as of today. And so the lesson from this example is that OGD is constantly working to streamline our recommendations so that we have similar recommendations for similar products. Now let's turn to the PSG for clascoterone topical cream. This was first recommended in November of 2021 with a comparative clinical endpoint study. And we worked with our colleagues in the Office of Pharmaceutical Quality to develop an IVPT method that may be used as part of a characterization-based bioequivalence approach. And as part of this method development, we optimized various parameters such as the apparatus, the receptor solution composition and volume, the sampling method and volume of sample, as well as the study duration. And so based on this research that we did in the lab, we were able to revise the PSG in August of 2023 to add a characterization-based bioequivalence approach that includes a recommendation for IVPT. I want to note that the OGD maintains a list of PSGs that we plan on publishing within the next 12 months. And this list includes PSGs that we plan to develop as new PSGs, as well as PSGs that we plan to revise. The key take-home point for this first part of my presentation is that OGD is constantly working to streamline recommendations across similar products within PSGs based on cutting-edge research. Now let's switch to when you might want to seek feedback from the agency and how to do that. So one aspect of your product development program where you might want feedback is on the formulation. For example, you may want feedback on the proposed concentrations of an inactive ingredient. The agency's inactive ingredient database, or IID, can be used to help justify levels of inactive ingredients in a proposed test formulation. And it's important to consider the context of use when selecting concentrations of inactive ingredients. For example, the route of administration, context of use, or duration of use, and patient population. But I'll note that while the route of administration is listed in the IID, the duration of use and the patient population are not. And so we strongly encourage you to discuss your proposed concentrations or your proposed formulation with the agency early in product development. And this can be important regardless of the bioequivalence approach that you intend to use. For example, before conducting a comparative clinical endpoint study, we encourage you to receive feedback on your proposed concentrations before going into the in vivo study. Another aspect where you might want feedback on your formulation is a no significant difference assessment. As I mentioned, I've previously given a presentation that focuses on this evaluation a couple of years ago. And in session 6 tomorrow, we'll have a presentation that discusses what should all be included in your formulation assessment submission. So in the interest of time today, I will just note that it's important to include all relevant information related to your formulation in your submission. I want to turn now briefly to TDS products. The PSGs for these products are very consistent in that they all recommend an in vivo bioequivalence study, an in vivo adhesion study, and an in vivo skin irritation and sensitization study. And in April of 2023, we revised this the guidance for industry on irritation and sensitization potential. And in that revised guidance for industry, we note that in some circumstances, an in vivo sensitization evaluation of a TDS product may be unnecessary if adequate justification is provided. And so if you want feedback on a proposed alternative study design compared to what's in the product-specific guidance for these products, we encourage you to submit communication to the agency through a controlled correspondence or a product development meeting request. You might also want feedback on your proposed bioequivalence approaches and your proposed study design. So if you're developing a product that is intended to be Q3 the same as the RS, we encourage you to submit a product development meeting request if no PSG is available, or if a characterization-based approach is not yet available in the PSG of the product you intend to develop. You may also want to send in specific questions about that you may have about the design or conduct of specific studies, such as the IVRT or IVPT studies. And Dr. Ghosh will be discussing these studies in more detail in session 6 tomorrow afternoon. Now, let's say you submitted a formulation for assessment as part of a controlled correspondence, and you get feedback that your proposed formulation may not qualify for a characterization-based bioequivalence approach under the Q3 sameness standard. But you would like to move forward with that formulation anyways. Then you may have a Q3 similar product, and we encourage you to submit questions about the studies that may be used to support this alternative approach in a product development meeting request. Finally, we encourage you to submit communication through a controlled correspondence or a product development meeting request if you intend to do a comparative clinical endpoint study, if the PSG is not available, or if you have specific questions about the design or conduct of the study. And finally, you may have noticed that in recent years, we've been adding this language to PSGs that indicate when an RLD product is a drug-device combination product, what the device constituent parts are, and what information should be submitted in your ANDA for assessment. However, I want to note that for topical products, we have not yet included this language into all of the relevant PSGs. And so if you have questions about whether an RLD product for a topical product is a drug-device combination product, we encourage you to send in a controlled correspondence to get clarification. And drug-device combination products will be discussed in more detail in session 4 this afternoon. But for topical products, a tube or a pump that does not alter the dosage form or does not meter the dose are considered to be container closure systems rather than devices. Alternatively, a pump that changes the dosage form, for example, from a solution to a foam, or that meters the dose, a TDS product that's meant to adhere to the skin, or a pledgette or swab is considered to be a drug-device combination product and would require comparative analyses of the user interface of the test product and RLD. So the take-home point for this final section of my presentation is the pre-ANDA program can be utilized to get feedback from the agency during both early and late-stage development, regardless of the bioequivalence approach you intend to use. So let's move to some challenge questions. Which of the following are considered to be an inefficient bioequivalence approach for topical products and is therefore prioritized for PSG revision? A, a comparative clinical endpoint study. B, a characterization-based bioequivalence approach. C, a VC study. Or D, a waiver of in vivo studies. The answer is A, a comparative clinical endpoint study. Which scenario would benefit from a pre-ANDA interaction with the agency? A, the PSG recommends a comparative clinical endpoint study, but you would like to use a characterization-based bioequivalence approach. B, it is not clear from the PSG if the product is a drug-device combination product. Or D, C, you would like feedback on the proposed levels of inactive ingredients prior to conducting a comparative clinical endpoint study. Or D, all of the above. The answer is D, all of the above. So in summary, the PSG recommendations provided by OGD are streamlined recommendations across similar products based on cutting-edge research and engaging with the agency through the pre-end program to gain feedback through product development can be beneficial regardless of the bioequivalence approach you intend to use. I'd like to acknowledge all of my colleagues at the FDA, and here is a list of resources that may be used for reference. So with that, I'd like to thank you for your attention. I look forward to addressing any questions that you may have during the panel session, and I'll invite Dr. Sacalozzi for the next presentation. Thank you.
Hello, everyone. Thank you, Megan, for the introduction. I would like to now take this opportunity to discuss a case study where we use modeling and simulation to enhance our understanding of structure-performance relationships for DAPs on topical gels. The objectives, learning objectives for today, I will discuss in silico methodologies for topical products that are applied on the skin. I will explain the data requirements and scientific considerations when building and validating dermal physiologically based pharmacokinetic, PBPK in short, models, in this case for DAPs on topical gels. And we will recognize how in silico models can be used to support regulatory decision making. The outline of the talk, Dr. Kelchen did an excellent job describing what is the rationale, the thinking behind developing product-specific guidances, what are the considerations. I will just highlight some relevant ones, relevant to the specific example I'm discussing. And then I will give a little bit of information on what are in silico methodologies for topical products. And I will spend most of the time talking about the case study, a dermal PBPK model that was developed for DAPs on topical gel, 7.5 and 5%. What were the considerations that went into the development, validation, and the application of the model? So PSGs for topical products contain harmonized recommendations that are in alignment with relevant general guidances, such as the Q3 characterization guidance, the IVRT studies for topical drug products guidance, and the IVPT studies for topical drug products guidance, all submitted for ANDAs. When the recommendations, when the PHGs contain recommendations within a characterization-based bioequivalence BE approach, then these BE recommendations are product-specific and they're based on an understanding of the microstructure and performance of the drug product. I believe that Dr. Kelchen made that clear. However, BE recommendations in the product-specific guidances may also include in vivo BE studies with PK endpoints or comparative clinical endpoint studies. So this talk will focus on a revision, PSG revision, that took place in February 2024. And this PSG revision refers to two products, the DAPs on topical gel 7.5% and the DAPs on topical gel 5%. Prior to February 2024, the PSG contained two options for this product. So for test products that met the no significant difference criterion, what was recommended was an in vitro characterization approach that required Q3 sameness and a comparative IVRT and IVPT passing studies. And this approach was supplemented by an in vivo BE study with PK endpoints. The option 2 was reserved for test products that did not meet the no significant difference criterion. After February 2024, this guidance was revised, and option 1 does not include anymore an IVPT study or an in vivo BE study with PK endpoints. The rest of the in vitro characterization studies under option 1 were maintained. This PSG revision, as Dr. Kelchen pointed out, was the result of our improved understanding on the product structure, microstructure, but it was also supported by modeling and simulation. This approach, the modeling and simulation approach, elucidated the relationship between drug product quality attributes which are the Q3 characteristics that are included in our PSGs, and the in vivo performance of the product. And these models that I will go into a lot more detail next establish the link between systemic and local dapsone exposure. So very, very quickly, the in silico methodologies that I will be discussing today are essentially mathematical statistical models that describe how the amount of dapsone in this case changes with time based on an administered dose. To generate this output, these models take into account information on the in vitro performance of the product. This could be, for instance, an IVPT study in the case of the dapsone gel. Information on the physiology, the skin, what are the physiological parameters that impact permeation through the skin and distribution throughout the entire human body. And also, these methodologies account for drug product characterization information. So a particle size distribution would be an example of this type of information. Now, for this case, we developed two models for these dapsone gels, different strengths, 7.5 and 5%. The objectives of these models were to understand the factors that are governing the permeation of dapsone through the skin, identify which drug product quality attributes that are specific to the dapsone topical gels may impact local and systemic bioavailability. And these models were finally applied to perform virtual bioequivalence assessments that evaluate the impact of formulation differences on BE outcomes. These virtual bioequivalence assessments were performed at the level of the plasma, so systemic exposure, following the application of the gels, and also within the context of an IVPT study, an in silico IVPT study. In order to develop these models, we leverage data on the in vivo permeation, the in vivo absorption of dapsone gels, and that is described through the model, schematic on the top here. But also, at the same time, we leveraged information from IVPT studies by developing an in silico IVPT model that really captured all the experimental conditions that are relevant to an IVPT study. These models work together to develop and validate each other and result in reliable, realistic predictions of dapsone permeation through the skin and absorption into the systemic circulation. So this is the general workflow that we followed. It is very similar between the topical gel, the 7.5 and the 5% strength. So we started with developing a systemic disposition model for dapsone. We leveraged literature data to do that. And then we started focusing more on the skin absorption component of that model and developed the dermal PBPK model for the 7.5% gel. We used data from approved ANDAs to optimize certain components of the model. And that optimized model then was validated against additional independent data sets that were extracted from other approved, additional approved ANDAs. Once this model was validated, we applied it towards virtual bioequivalence assessment for all ANDAs that were considered for development and validation. And in addition to the plasma BE assessment, we also were able to perform bioequivalence assessment in the stratum corneum and the dermis for this product. What was also unique in this specific case is that we were able to validate the model using IVPT data. And in my next slide, I will go into a lot more detail on this. The workflow for the development and validation of the dapsone topical gel 5% model is very similar. We started with the same systemic disposition model. And now we switch to ANDAs, approved ANDAs for the Dapsone topical gel 5%. We saw that the model for the 7.5% topical gel performed really well. So all the data from the approved ANDAs for the 5% was considered external validation. We did not need to optimize the model at all for the 5% strength. And similarly to what we did earlier with a higher strength, in this case, we applied the model for VBE assessments for the ANDAs, approved ANDAs for the 5% strength in the stratum corneum and dermis, so locally and systemically. Now, we just discussed the workflow for the 7.5% gel, and I said that we used IVPT data to further validate this model. So in parallel to this specific in vivo model, we developed an in silico IVPT model, which described the IVPT study conducted for the Dapsone topical gel 7.5% within an approved ANDA. We optimized certain parameters of the model. I will describe those in my next slide. And we used that partially validated IVPT model, in silico IVPT model, to assess by equivalence between the RS and the test product in the skin, the stratum corneum, and also the receptor media. So this is a schematic that will help you understand how these models are built. They're essentially mathematical equations that describe how the API is partitioning, is being released from the formulation, which is the gray compartment here, into the stratum corneum and the rest of the skin layers. In order to describe mathematically this process, we can account for Q3 aspects of the product, such as formulation pH, particle size distribution, apparent viscosity, and solubility. So these are direct inputs into the model, and there's information that is collected within the scope of the Q3 characterization of the product. Nothing really is required in addition to those Q3 studies. We do account for formulation composition when we are building these models. As I mentioned, the Q3 aspects, the drug product quality information is included into the model. And for validation, we used in this case in vivo systemic PK data and IVPT study data. We made certain assumptions regarding the evaporation and drying rate of the gels, but this can also be characterized experimentally. And in the case of the in vivo application, we did not assume any rubbing, although this was reported in the clinical study protocols. So we utilized these models for VBE assessments. We performed 10 virtual BE trials where we matched essentially the in vivo BE studies that were conducted in support of the approved ANDAs we worked with. And we assessed by equivalence virtually for Cmax and AUC in the stratum corneum, the dermis, and the plasma. In terms of the IVPT studies, we again matched the conditions of the IVPT study in terms of donor demographics, replicates, and dosing conditions, and the experimental conditions. And we assessed B for maximum amount and AUC in the stratum corneum for Dapsone and cumulative amount and Jmax of Dapsone in the receptor solution. This slide summarizes the performance of the model, where we see that overall the blue lines that are the model predictions overlay relatively well with the observed data, which is the black dots. Now, when it comes to IVPT data, we developed an IVPT model, as I mentioned earlier, and in this case, what we see, the model predictions are the red and blue lines, and the observed data are the squares. What was interesting here, and I would like to note, is that what we saw is that when we include variability in the applied formulation on the receptor, on the donor side, we were able to describe the variability that we see in these type of studies very well. So that's an interesting point to consider in the future. Similarly, in the case of the 5% gel, we're summarizing here the performance of the model, predictions and observed data overlay relatively well. So these models were applied to assess bioequivalence between the RS and the test product in the stratum corneum, the dermis, and the plasma across all the ANDAs that we utilize for model development and validation. And we saw that when we have a test product that meets the no significant difference criterion, Q3 sameness between those two products leads to BE locally and systemically. And certain Q3 attributes, in this case particle size distribution and apparent viscosity, impact BE outcomes and should be closely controlled. Same were the outcomes in the case of the virtual IVPT studies, where we looked at Jmax and the cumulative amount in the receptor solution and also amount of dapsone in the stratum corneum. All of these ANDAs were considered, were found to be bioequivalent, and similarly to the in vivo study, this modeling and simulation approach showed that Q3 sameness between the reference and the test leads to BE locally and the receptor solution. So in conclusion, this modeling approach improved our understanding of the structure-performance relationship of dapsone gels. Apparent viscosity and particle size were shown to be important Q3 quality attributes. And these developed models can serve as tools supporting a risk-based assessment. The compelling evidence from this PBPK modeling approach aligns well and reinforces the BE recommendations in the aforementioned draft PSGs for other topical gels, as Dr. Kelchen pointed out. So first challenge question: which Q3 attributes were identified as impacting bioavailability based on the developed dermal PBPK model? Formulation pH, rheological properties, particle size distribution, and droplet size distribution. And the answer is B and C. Challenge question number 2. Enhanced understanding of the structure-performance relationship of dapsone gels gained through the PBPK modeling research led to a successful product development meeting for an applicant, an ANDA approval, or a PSG revision. And the answer is a PSG revision. Thank you very much for your attention. I would like to thank everyone, my colleagues at DTP1 and DQMM, for their support on this project. And we can move on to the next talk that will be delivered by Dr. Tanaz Ramizanli on the approaches for evaluation of formulation differences on performance of topical products. Thank you.
Good morning, everyone. This is the last presentation in session 2, and I'm going to talk about the approaches we have been working on to evaluate the impact of formulation differences on performance of topical products. The objectives for my presentation are first to describe consideration for formulation development of a test product that does not meet the no significant difference criterion. Second, to evaluate the impact of compositional differences or Q2 differences on bioavailability of topical gels. And the last is to evaluate the impact of Q2 changes on sensory perception of topical gels. So you heard from Dr. Kelchen's presentation earlier in this session about the bioequivalence approaches that are currently recommended in our published PSGs for topical products. In this diagram, you can see the potential strategies for BE that was developed or are under development under GDUFA Regulatory Science Program. And they have grouped them based on the requirement in the for the formulation. So when a test product, for example, meets the no significant difference criteria and it is pre-qualified for a characterization-based approach, and this approach is currently recommended in many of our PSGs for topical products. But when there are differences in the formulation of a test product compared to reference standard, then there are other approaches that we have been working on to develop for this category of test formulation. So that there are approaches based on cutaneous pharmacokinetic studies, such as ones using dermal microdialysis or dermal open flow microperfusions that are in more advanced stages of development. And there are others that are in earlier stages of development, and we are still investigating them, such as cutaneous PK studies using imaging or spectroscopy-based tools, or the ones that is the focus of the today's presentation is to evaluate the impact of formulation differences on thermodynamic activity and sensorial perception of topical products to be able to expand the characterization-based approach to some of these test formulations that may have compositional differences compared to the reference standard. So we first needed to understand the function of excipients and their impact on thermodynamic activity of the drug in topical formulations. And we started with single-phase gels, which are the least complex form among the semi-solid topical dosage forms. And in the figure, you can see different types of excipients that may exist in a single-phase gel. We have solubility modifiers such as propylene glycol, polyethylene glycol, or glycerin. We have gelling agent, preservative, pH modifier, solvent, etc. And among these different types of excipients, we identified that probably the Q2 changes in solubility modifiers may have the highest impact on thermodynamic activity. So we started focusing on the solubility modifiers in our research. And in order to predict thermodynamic activity changes, we first needed to define fractional solubility, or alpha, which can be calculated from concentration of solute divided by saturation solubility of the drug or solute in the solvent. For example, if we have PEG water formulations, by increasing the amount of PEG in the formulation, since it is a solubility modifier, the solubility of the drug of interest most likely would increase. For example, here in this example, the saturation solubility of this drug has increased by increasing PEG concentration. But if we have a constant drug concentration, according to this equation, by increasing the denominator, the fractional solubility would decrease. So by increasing PEG concentration, the saturation solubility increases, but at 7.5 milligram per ml concentration, we have the lowest alpha with the 40% PEG formulation. And fractional solubility is often predictive of the thermodynamic activity of the drug in the formulation and how likely the drug molecules would leave the formulation and partition into the skin. So one of the earlier studies that our collaborators did was to make series of propylene glycol water formulations. These were like 2-ingredient formulations, and the saturation solubility of diclofenac sodium, a lipophilic drug, was measured in series of this formulation with variant amount of PG from 0% to 100%, and as expected, increasing in PG amount, saturation solubility increased. Then several of these formulations were selected in the range of 10% PG to 50% PG, and they conducted an infinite dose IVPT study. So as also explained in the previous slide, when we have a constant drug concentration, for example, in this case, 2% diclofenac sodium, by increasing the amount of solubility modifier, we expected to see a decrease in the thermodynamic activity. And the IVPT results also show that with 50% PG, we got the lowest flux, steady state flux, and with 10% PG, we had the highest steady state flux. I would like to know that since we had infinite dose in this IVPT studies, the metamorphosis of the dosage form was negligible, but when the topical product is actually applied on the skin by human or by patients, the finite dose is used, which would undergo metamorphosis and drying, and following solvent evaporation, there may be changes in drug solubility at the application site, or even change in microstructure and Q3 properties, for example, change in rheological properties of the formulation as the formulation dries, and all of them may lead to change in drug permeation and bioavailability. So while we can measure fractional solubility at time 0, we didn't know how this fractional solubility or thermodynamic activity would change across the product application on the skin. So what we did that our collaborators, they monitor the drug solubility or saturation solubility at the application site by measuring drug concentration in a donor compartment. So they basically had the Franz diffusion cell set up similar to how the IVPT is set up, and they used a semi-finite dose on the skin in the donor compartment, but instead of sampling the receptor compartment, they sampled the donor compartment and measured drug concentration in the donor compartment at different time points. So using this methodology, we would be able to have fractional solubility profile across the duration of application here, for example, for 6 hours. And here shows the results we got with HEC gel formulations, which included PEG 200 as a solubility modifier at different concentration, 1%, 10%, and 20%. And here the drug model was diclofenac sodium and with a fixed concentration. So as is shown here, with different amount of PEG, we had different fractional solubility, and during the course of 6-hour application, the fractional solubility with 1% PEG increased, and for the other two it kept constant. But what is important is we saw a well-separated profiles from the 1% and 10% and 20%, which suggests that the thermodynamic activity of the drug in these 3 formulations are different, and subsequently the IVPT results also showed that the permeation rate was significantly different from for the diclofenac from the 1% PEG formulation compared to 10% and also 20%. The same similar experiments were conducted with HEC gels that contained metronidazole. So this is a hydrophilic molecule with different pKa and different physicochemical properties. And here, interestingly, with the variation in amount of PEG from like 1% to 10%, 20%, did not result in significant differences in fractional solubility at time 0 and also during the course of 6 hours. So subsequently, the permeation profile was also very similar for these for metronidazole from these 3 formulations. So we were able to establish a nice correlation between the drug bioavailability and the thermodynamic activity. Next, we wanted to focus on smaller Q2 changes and their impacts on bioavailability. So here shows examples of gel formulations where PG, propylene glycol, was used as a solubility modifier for both metronidazole formulations and diclofenac sodium formulations. And we first evaluated the saturation solubility of the drug in different amount of PG. And we, for identify the reference product, we selected portion of this curve where it is relatively steep and it's within the range of PG amount used in approved topical products. So for metronidazole, the reference formulation contained 30% PG, but for diclofenac, the reference formulation contained 40% PG. And then from there, we calculated the PG amount in plus minus 10% difference and plus minus 25% difference in PG, and make those formulations with metronidazole and diclofenac. So the fractional solubility measurements showed that with plus minus 10% variation in PG, the thermodynamic activity is not changing, and subsequently the permeation rate is not changing compared to the reference formulation. But with plus minus 25% difference, the fractional solubility has changed, and the flux profiles also are distinguished from the one for the reference formulation. With diclofenac sodium formulation, while we see some separation of for the plus minus 10% formulation than compared to the reference, the IVPT result did not show significant difference in bioavailability of diclofenac for the plus minus 10% variant formulations and the reference formulation, but the profiles for plus minus 25% variant formulation were different compared to the reference formulation. Next, we wanted to understand whether the Q2 changes in topical products would have other impacts beyond changing the bioavailability. For example, would differences in Q1, Q2, Q3 of topical products result in differences on how the product would perceived by human subject and potentially change in therapeutic equivalence. So for that, we needed to establish a correlation between Q2, Q3, and sensory perception of topical products. And also we needed to understand whether the characterization of Q3 properties, such as rheological behavior of topical products, predict the sensorial differences that may be perceived by subjects. So for that, our collaborators did develop objective tools to measure some relevant Q3 attributes and also conducted in vivo sensory panel tests of those formulations to be able to correlate these Q3 with those sensory attributes measured. So here we also started with single-phase gels, and we identified relevant sensory attributes of those gels. In the left image or figure shows the list of sensory attributes that we evaluated for topical gels, like spreadability, smoothness, shine, cooling sensation, etc. And we identified which of them potentially could have more impact on therapeutic equivalent. So for this study design, after identifying the sensory attributes, we needed to find for each sensory attribute what would be the relevant Q3 attributes that we can measure in vitro, and how we can measure that using what kind of instrumental technique, and also potentially what formulation variables can more impact the changes in these Q3 attributes and the sensory attributes. For example, for something like a cooling sensation, potentially a drying rate study was assumed to be a relevant test to inform about the cooling potential of a formulation, and probably the change in amount of solvents or co-solvent may have an impact on changing these sensory attributes. And for something like spreadability, we assume that rheological behavior of the gels, like measurement of zero shear viscosity, yield stress, etc., with a rheometer would inform the change in the spreadability, and potentially the amount of gelling agents would have the highest impact in changing these Q3 attributes and this sensory attribute. So here are the results for the Q3 test. Some of them are shown here, and also the in vivo panel study for these three attributes that are shown here. For several carbopol 980 gel formulations that were made with different composition of a gelling agent and alcohol. And in this figure, the data is normalized for each unit, for each attribute, in the range of 0% dark blue up to 100% yellow. So formulation 2 here was the reference formulation. And for example, in formulation 3 and 1, we had differences in the amount of carbopol. Formulation 3 had like double the amount, and formulation 1 had half of the amount used in formulation 2. And these changes in the gelling agent resulted in significant change in the zero shear viscosity for these two formulations. But only in formulation 3, we observed a significant difference in the perception of stickiness and spreadability compared to formulation 2. And in formulation 4 that we had alcohol as opposed to formulation 2, the slipperiness of this formulation was perceived significantly differently. The similar studies was done with hydroxyethyl cellulose gel with different compositions of PG and propylene glycol and alcohol. Here, formulation 3 was the reference formulation, and the variation in amount of alcohol in formulation 1 to 4 resulted in significant change in zero shear viscosity. And in formulation 2, we had significant difference in perception of slipperiness. In formulation 4, where both the amount of propylene glycol and ethanol was different compared to reference formulation, the stickiness of the product was also felt differently. In conclusion, this preliminary results for this gel suggest that when we have Q2 changes, we may or may not see significant differences in sensory attributes. But when we have significant differences in Q3 attributes, we likely would see some, or we may see, or it's more probable that we see differences in sensory perception of those products. So with that, I summarize that FDA is investigating alternative scientifically valid methods, including in vitro approaches to support assessment of BE for topical products that have compositional differences compared to reference standard. The current research data suggests that when there are differences in thermodynamic activity, such differences may result in bioavailability as shown as evaluated by IVPT. The Q3 properties assessed instrumentally in vitro may be valuable in understanding most of the sensorial differences for topical gels in vivo. And the current data suggests that large differences in Q3 attributes, such as for like neurological, tribological, or textural properties, would likely perceivable to human subject. And finally, research is underway to further evaluate the impact of Q2 differences on bioavailability and product perception of topical gel formulations. So here are my challenge question. The first question is, which statement is not correct? Q2 differences would always result in changes in performance of topical products. B, it may be feasible to assess fractional solubility of the drug in conjunction with IVPT to assess impact of Q2 changes on performance of the product. C, significant Q2 differences may result in changes in performance of the topical product. And D, metamorphosis of the formulation following topical application may change the microstructure of the product. So the correct answer is A, because Q2 differences would not always result in changes in performance of the topical product. Then second question is, which of the following Q3 attributes is more likely to correlate with spreadability and stickiness of topical gels was based on the data I presented? A, friction of coefficient, B, zero shear viscosity, C, drying rate, D, all of the above. And the correct answer is zero shear viscosity. So with that, I would like to acknowledge and thank my colleagues and supervisors at US FDA and our research collaborators. And thank you very much for your attention, and we'll be happy to answer to your questions during the panel session. Thank you very much.
Thank you very much. We call our speakers back to the panel table, and we would like to welcome two new panelists, Dr. Hiran Patel, Senior Staff Fellow from the Office of Bioequivalence at OGD, and also Dr. Pahala Simamora, Division Director of the Office of Product Quality Assessment in the Office of Pharmaceutical Quality. So we'll do the same thing like we did last time. Any in-house questions, please come up to the microphones. We have such a lovely panel of experts here. They're just waiting to answer your questions. Brave, sir. Yes. I hope you're going to the microphone. Okay, yeah.
Yeah.
All right, question at mic 1, please.
Marie Ducharme, Learn and Confirm at University of Montréal. I have a question for Dr. Tsakalozu regarding, first of all, it's a comment, and then the other one is a question. The comment is, you said that the model established a link between systemic and topical concentration. Isn't that an assumption of the model?
It characterized the relationship, right?
Well, it's an assumption of the model. It's built in.
Yeah, it's part of the structure of the model. Yes.
Yeah. Okay. So I think the sentence is a little bit misleading. I mean, just a comment.
Sure. Thank you.
The question is, what is your validation criteria? You know, you showed, you know, validation with data and all of that. What is it?
Yeah, that's an excellent question. So you're asking this question because the performance of the model was not always ideal, right? So the challenge that we typically see in PBPK modeling, and it was very, very obvious with this type of work that I presented here was that there was a lot of variability in the in vivo PK data. The conditions, the study conditions varied between the ANDAs that were utilized for model validation in terms of the population, the application sites, the doses that were applied, and the conditions. So rubbing, for instance, is a big consideration that unfortunately currently we don't take into consideration into the model. And this is a condition that may impact skin permeation to a great extent. The other challenge specifically for this work is that the application site was not the same across all of these studies. So that introduced variability in our model predictions. The criteria that we used in terms of the internal validation is a 20% difference between the predicted and the observed. We want to be as close as possible when it comes to the internal validation, but for data sets that are used for external validation, there are parameters that come into play in terms of the population variability that we cannot always account into our model, and that leads us to the model performance that you saw here.
Yeah, but what was the criteria? So it was like minus to plus threefold, or what was it?
It was twofold.
Twofold? Mm-hmm. For the mean?
Yes.
The mean being twofold. Okay.
Yes. Thank you. Mm-hmm. Thank you, Dr. I'm going to get this. Dr. Sakalaka Lusu. I'm just saying thanks. Okay. Any in-house questions? Any? Okay, let's go online, please.
Okay. We have actually another question for Dr. Eleftheria Sakalozu. How are in silico models being validated for a given drug product and indication? If the same model is being used for another product, what variation will have to be considered?
Thank you. That's another good question. So the models that mechanistic models, such as the one that I discussed today, are drug product specific. So they're built utilizing data that are specific to the product of interest, and then they are validated either with IVPT, microdialysis, open flow microperfusion, or in vivo PK data that are also collected after following application of that specific product. So in other words, in the example that I described here today, for every ANDA, two models were developed, one for the reference standard and one for the test product. The validation is performed across all the available data sets. Obviously, we're looking for data sets that are good quality, that are collected under controlled studies. And against the previous question, obviously there is a variability around those data sets. I hope that I've answered this question. Hopefully I didn't miss anything. Thank you.
Thank you. Next online question, please. We'll just continue with the online unless somebody comes up to the mic.
Next question is directed to Dr. Pahala Simamura. Could you please elaborate on the usefulness of in vitro characterization and in silico approaches to support SUPAC changes?
Thank you. That's such a good question. As we know, SUPAC guidance has been in place since in the 90s, so it's been a while. It's been evolving so far, as Dr. Megan Kelchen presented, all the evolvement to date. A lot more we learn about drug product, whether it's simple or complex, and so on and so forth, right? So, and also, as the previous section mentioned about looking at the totality of the data as well. The point is, I think with a strong product development, strong product understanding and process understanding, I think it would make it easy when post-approval we want to make changing to certain thing. Is there like a specific things to do? Maybe not. Maybe it's a combination of things to look at the totality of the information. I don't know if I hope I answered the question. I know it's quite generic, but compared to the what we have which is currently is the standard for post-approval change. But I think now we have identified a lot more parameters that are more meaningful for to identify to mitigate the risk of changing certain things post-approval. Thank you.
Thank you. Next question, please.
Next question is for Dr. Megan Kelchin. Do I need to get feedback on a proposed concentration in my test product if the level is below what is listed in the inactive ingredient database, or IID?
Thank you for the question, and thanks for the opportunity to clarify this. So the external IID is a great starting point when developing or trying to figure out which concentrations you want to use in your proposed test formulation. Because as I mentioned in my presentation, the IID includes the route of administration for the drug product. So the idea being that your proposed concentration should be below the level that's included in the inactive ingredient database for that route of administration. But as I mentioned, the context of use, so for example, the duration of use or the patient population that product is used on or used in is not listed in the IID. And so what we don't want to do is use a product that is for an acute indication to justify the levels of a product or a concentration that's to be used in a product for a chronic indication. And so while it's not necessarily required, we strongly encourage you to come in with your proposed concentrations and get feedback from the agency. And again, this can be helpful even if you want to do an in vivo study, because we don't want you to do a comparative clinical endpoint study just to find out your proposed concentration is too high compared to what we have in-house. Thank you.
Thank you, Dr. Kelchin. Next question, please.
Next question is for Dr. Hiren Patel. Could you please highlight the criteria for recommending IVPT in the in vitro characterization BE approaches? Also, how can an applicant interact with the agency for feedback and recommendations?
Sure. Thank you for the question. It's an excellent question. As we heard Dr. Kelchin talked about how the characterization-based BE approach laid out, it based on the complexity of the drug product. For example, and it is, as we heard in the earlier session too, that it's a weight of evidence-based approach. So it is how we systematically mitigate the risk of failure mode. For example, if the drug product is a single-phase formulation, then if the formulation is meet the no significant difference criteria, which is based on the Q1, Q2, have similar Q3. And if you conduct IVRT study, we believe that it systematically mitigate the risk of failure mode. There are certain exemptions. For example, if it has a complex inactive ingredient like white petrolatum, then we may recommend IVPT study, even though it's a single phase formulation. However, if the formulation is a multiphasic, we typically recommend IVPT study in addition to the Q1, Q2, Q3. However, there may be some exception on multiphasic formulation. For example, if there is no clinical relevance, we may waive the IVPT study. So I hope that answers the question about in which instances we recommend IVPT study. The second part of the question is if you seek agency's feedback either on the utility of the characterization-based approach, I would encourage you to submit product development or control correspondence. Thank you.
Thank you, Dr. Patel. We do have an in-house question, please. Go ahead.
Yeah, I have a practical question. Let's say if you use topical drug applied to skin and for a certain time, and you continue measure plasma concentration on those things. So there's a timing difference beyond how when you stop use drug. When you modeling to the validation and how long time you need to take after drug finish.
That's a question for me.
Yeah.
Okay. Yes. So the model predictions are considered validated for as long as there is observed data to track, to compare those simulations against the observed data. So if you have a PK, an observed PK profile that is 12 hours, but your model is predicting for 48, clearly the part of the model predictions that are not validated against, they're not compared against observed data, are not as credible as the ones that are compared to actual observed data, right? There are, when we are assessing model validation, when we're assessing model performance, we're looking for the overall shape of the curve we're interested in, right? So Cmax, AUC, Tmax, and then additional characteristics that may be relevant to the product. Multiple peaks, maybe.
Can I follow up?
Please.
So in certain cases, some drugs, they have a faster metabolism rate. And even when they continue release, you might see drug in skin tissue, but the plasma concentration can go down necessarily to 0. And in that situation, you don't see too much drug concentration in plasma, but you still know drug is releasing and you see in tissue. And a validated modeling is going to be what timing cutoff. And even we should use plasma to do the modeling at all. Just wanted your thought about that. I don't know what's right or wrong, just your thought.
Yeah, the case that you're describing is definitely challenging. It would be very useful in this case to follow a more mechanistic approach, modeling approach, I mean, where there's additional information that goes into the model that can justify, that can support further the model predictions. Then it will also be helpful to show that this same model or similar model or modeling suite is also behaving well with other products. So the uncertainty that is present in your own case is in some way mitigated by accumulating knowledge in other cases. Additional areas like matrices where the model can be validated would also be useful. Additional sources like multiple studies will also be validated, will also be useful. Best practices when we are developing a model, overall best practices present in our guidances should be followed to kind of mitigate, to increase our confidence and mitigate that uncertainty, that risk that we have with the lack of the data. It's a very general question.
Thank you very much for that response. Online, please.
We have an online question for Dr. Tanaz Ramzanli. How does comparing metamorphosis and thermodynamics translate into a patient's or prescriber's concern about similar look and feel of a topically applied dermatologic drug product?
So as I shown in the presentation, one of actually our research project focuses on that and to see how the differences in Q composition and Q3 properties can change the sensorial perception of topical products. So our early results or like preliminary results suggest that we can correlate these sensory attributes with in vitro Q3 tests. And our in vitro Q3 tests are usually more like strict to differentiate compared to how the differences will be significantly felt by patients. So, so eventually, I think if we have differences in the composition of the formulation, and the formulation is not gonna go through the clinical endpoint study where it's tested in patients, there might be alternative ways that we can mitigate this failure modes, and we are working to develop the alternative studies for that.
Thank you. Next question, please.
Next question is for Dr. Sakalozu. Could you comment on whether the US FDA develops models or methods for assessment of in silico studies, or prefer that the manufacturer do so?
Thank you for the question. It really depends on what is the need that a product presents, an application presents. There are, if an applicant identifies the need for their product to be supported by in silico approach, we welcome this. We will call it alternative B approach, and we will be we'll provide the necessary feedback. In cases where we feel that there is a risk with the product, in cases where we want to inform our regulatory policies, such as the guidances, we would conduct internally modeling and simulation. So both.
Thank you. I think we have time for one more online.
This is a general question for the panel, or for anyone on the panel. PSG revisions are a big headache for industry. Therefore, why don't you publish a set of correct studies to begin with, rather than revising them one at a time based on approved ANDAs?
So I'll start, and others can chime in. So the recommendations we have for in each PSG is product specific and is recommended based on our evaluation of the data for that product and understanding the complexity of the product. So we definitely cannot and would not recommend a general studies for series of different products, because each product, each guidance or product specific guidance is specific to that particular drug product.
Thank you. Do you want to squeeze in one more online?
Can I actually add something?
Sure.
I'll note that when we publish PSGs for newly approved drug products, sometimes we want to do some additional research before we publish, say, a characterization-based approach. And so we might publish a comparative clinical endpoint study as part of the PSG to get the guidance out there, to give industry an opportunity to have a PSG available. But there might be some internal research that we're doing, such as like the clascoterone cream that I discussed in my presentation. Thank you.
Okay, I think we'll call it, we're just on 11:45. So thank you to our panel. We will break now for lunch. You have an extended lunch. Please be back at 1 o'clock. Please do your QR survey for graphics. Please look at the posters. And if you come back on time, we're going to have a trivia question with a little gift bag for the winner. So have a good lunch. Thank you.
Okay, everyone, 1:30. We are ready to start. And I made a promise, right? Anybody remembers? We're going to have a trivia question. Let's go. Ooh, alrighty. So SBIA, Small Business and Industry Assistance, we have been doing this since 2010. We have an online repository named SBIA Learn. Anyone familiar with that repository? Online, anybody? I can't see you online, but please say yes or no. And online attendees, my apologies, but this, little gift bag is only for in-house people. So SBIA Learn, like I said, and we actually did some Spanish modules specific to generic. Stay right there in the blue box for me, please. Up a little bit. We actually did some Spanish captions. We have 10 modules in Spanish specific to the generic industry. So it's available there as well. We have 10 more that we have to put up. And it's an ongoing project that we're working with. And if you scroll down, you see you have SBIA recordings on YouTube, upcoming SBIA events. You can search through the type. Jeff, do you want to do a little click on the arrows for me? So whether it's on demand, because we do some of those webinars as well, on demand, symposium, webinar, etc. And then if you do topic, you can have it by, various categories. And we're in the process of updating this. By the way, did anybody take the graphics survey? Please, not yet. Please take the graphics survey. We need you guys to take the graphics survey so we can better get our word out to you in a manner that you can better digest. All right, we'll forget about that for now. So this is the way you can get our webinars, symposiums. Every webinar, like each session of these 2 days, morning, evening, will be condensed to YouTube postings, and we post that up there for easy reference. The trivia question. How many, and whoever gets the closest answer, how many of these YouTube posts do you think we currently have in this SBIA Learn report? All right, we're going to disqualify that answer. Well, now we lost it. All right, 817. So that's the answer. And Nora, you will give this gentleman our prize, please. All right, thank you guys for playing along. So this is SBIA Learn. We have everything from 2017 there. So please go back. All our generic drug forums, all our past 7 years of advancing generic drug development, all of those are there. So please utilize it. Okay, let's begin our session 3, Research to Support Guidance Development for Inhalation Drug Products. Our first speaker will be Dr. Liang Feng Hang, who is a clinical analyst from the Office of Research and Standards within the Office of Generic Drugs. Then we'll have Dr. Ross Wallinger, who is our senior chemical engineer at the Office of Research and Standards within the Office of Generic Drugs. And then the third speaker we will have is Dr. Changning Goa, who is a supervisory chemist at the Office of Pharmaceutical Quality Research, OPQR, within the Office of Pharmaceutical Quality. So let us welcome our first speaker, Dr. Liang Feng Hang. Thank you.
Hi, good afternoon. I'm Liang Feng Han. I'm the clinical analyst at the Division of Therapeutic Performance 1, Office of Research and Standards, Office of Generic Drugs. I will start this session by discussing FDA's general considerations using the alternative B approach in lieu of comparative clinical endpoint B study for suspension-based metered dose inhalers. This presentation has 3 objectives. First, I will recognize the challenges with conducting the clinical BE studies for orally inhaled drug products. Then I will discuss the available tool and the data from FDA-funded research and the external inputs that contributes to our development of alternative BE approach. Finally, I will list some recent PSGs for suspension-based MDIs that recommends alternative BE approach and discusses their study design considerations. For local acting MDIs and DPIs, FDA traditionally recommends an aggregated weight of evidence to establish BE that includes a clinical BE study. But we know there are many challenges for the clinical BE study. For example, these studies has high variability and the lower sensitivity due to the flat exposure response. As a result, these studies usually requires larger samples and a long study duration. Consider the huge cost and the long time required for conducting these studies. Now FDA considers an alternative BE approach in lieu of the clinical BE study that includes in vitro, in vivo, and in silico methods. Ideally, the alternative BE approach could assess each step of the local drug delivery from pre-actuation to the local deposition. These steps include the formulation and the device characteristics before actuation. And after actuation, the aerosol is formed. So after the aerosol leaves actuate, the droplet size changed due to the propellant or other excipients evaporation. After the aerosol enters airway, how the patient's anatomy and the physiology condition affect the local drug deposition. So after the final drug local deposition, the dissolution and the absorption from the local site. And we now have many in vitro studies, such as realistic APSD, dissolution, and MDIS that can provide the information for each step during the process. But we have limited in vivo study methods and some in silico study methods that provide information on some of these steps. And the alternative BE approach was first introduced in the PSG for the solution-based MDIs, like Cuba and the Cuba radiator. The PSG states that if the test formulation is Q1, Q2, same as the LD formulation, and that the test device is similar to the LD device, the end applicants can take an alternative BE approach that includes characterization of emitted sprays, morphology imaging comparison, and the realistic APSD dissolution study, and the in silico modeling, and the alternative PKB studies. And after that, the next question is that can we use the same approach for the suspension-based MDIs and DPIs? To answer this question, FDA hosted a 2-day workshop that invited experts from industry and academics. The workshop consisted of the larger conference and the small group discussions. At the workshop, FDA presented the thinking on the alternative BE approach and received feedback from the external experts. The consensus at the workshop was, in general, the alternative B approach for the solution-based MDI could also be used for the suspension-based MDIs and the DPIs. But some studies, like the realistic APSD dissolution in silico methods, are more useful to establish B than other studies. And the particle morphology charcoal block PKB studies are partially useful or for the confirmatory purpose. Evaporation rate and the velocity profile evaluation, pre-actuation characterization of the formulation have limited utility. It should be noted that the alternative B approach is product specific. That means not a single alternative B approach that works for all the products. So after the workshop, the FDA compiled the outcomes from the event and posted the new PSGs for the suspension-based MDI. All these PSGs take the option-based approach. If the test product, a test formulation is Q1, Q2, the same as the RD formulation, and the test device is similar to the RD device. Oh, that the option one includes that the conventional in vitro B studies, the in vivo PKB studies, and the alternative B approach include additional in vitro B studies, characterization studies, and the charcoal PKB studies, and the in silico studies. If the test formulation is not the Q1, Q2 as the RD formulation, and the test device is similar to the RD device, and the end applicants can take option 2 that follows traditional B recommendations that include a clinical study. And the option-based approach was first introduced in the PSGs for generics referencing Bavaspi aerosphere and the Bradstreet aerosphere. There, FDA approved the suspension-based MDIs indicated for the COPD treatment. Their formulation contains this DSPC and the calcium chloride, and these components contributed to the formation of the phospholipid-based porous particles. FDA posted the 2 PSGs for Bavaspi and the Bradstreet aerosphere in this February, then posted 4 PSGs for other suspension-based MDIs this August. For option 1, the test formulation, as I mentioned before, should be the Q1, Q2 the same as the RD formulation. The in vitro B study included the conventional in vitro B study and the 2 additional in vitro B studies that are realistic APSD and the dissolution. Comparative characterization studies, such as particle morphology of the emitted dose, the in vivo PKB study, and with and without the charcoal block. And the in silico modeling is optional for the B establishment, and the test device should be similar to the RD device. As I mentioned before, the alternative B approach is product specific. You can see here is the dissolution study is only recommended for three products with slow dissolving APIs. And the morphology study, particle morphology study, is only recommended for 2 products that can have with the porous structure. For option 2, the test formulation could be Q1, Q2, or not Q1, Q2 to the RD formulation. The in vitro B studies contains the conventionals at the in vitro B studies, and the comparative characterization study including the particle morphology, and the in vivo PKB study without chalk block, and include a clinical BE studies. And the in silico modeling is also optional for BE establishment. The test device should be similar to the RD device. And you can see here that the option 2 is the same for all the products. Okay, now I will discuss the study design considerations for each alternative BE study. The GDUFA funded research outcome indicates that there are many factors that affect the APSD study. For example, the mouse throat model size, the inhalation profile, and there's other factors including the filing time point, and the angle of the mouse throat model inserted. And we also noticed that the response to the various study factors is product specific. And in this figure shows the fine particle fraction variability. It's that for the different products, when different size of the mouse throat models were used in the realistic ABSD study. Therefore, we recommend that your method development should consider the mouse throat types and size and inhalation profiles and other factors. This figure shows that the different mouse throat models and the realistic inhalation profiles and the constant flow rate inhalation profiles. We recommend that the realistic APSD can be conducted at the beginning life stage. If the conventional APSD study shows that no difference between the beginning stage and the end stage. And the applicants also can take a bracketing approach. For example, the small and the largest size mouse throat models plus weak and strong inhalation profiles. As long as the combination of the mouse throat models and the inhalation profiles can cover the expected inter-subject variability of the indicated patient population. Alternatively, you can justify your choice by showing good in vitro, in vivo correlation if you have this data. And the inhalation profiles should be obtained from patient. And we see some submissions that they provide the inhalation profiles from the health subject, and we think it may not be appropriate. And the B is based on the PBE analysis of the impact size mass for each mouse throat model and the inhalation profile combination. And the FDA welcomes that alternative statistical approach if this approach can be scientifically justified. And you can also request a pre-end meeting request to discuss your proposed statistical approach. Post dissolution study, we also found that many factors can affect the dissolution study, especially the particle size of the emitted particles. And we noticed that currently there is no standard methods. And the method development could be product specific. And in this figure at the bottom, you can see that this figure shows a good dissolution study, because this dissolution study can distinguish the different dosage form and the different particle size. And we also acknowledge that the need for the dissolution study is product specific. For fast dissolving API products, the dissolution study may not be necessary. And the PSG recommends the dissolution study should be conducted at the beginning of the life stage. And the study should collect the aerosolized dose of similar drug mass between the T and the IS products. And your method should be optimized and validated by modulating your apparatus sample collection and the dose, etc. And your dissolution study must be discriminatory, that can identify the difference in the deposited drug particle size. And the B is based on the comparative analysis of dissolution profiles with inappropriate statistical methods such as F2 analysis. And after inhalation, some OIDPS could be swallowed into the GI contract. If there is a significant GI absorption from GI contract, the conventional PKB study without charcoal block cannot distinguish the systemic exposure from the inhalation portion or the swallow portion. Therefore, for these products, we recommend a PKB study with charcoal block that can eliminate the GI absorption and obtain the PK profile only from the local lung deposition. And we understand that the charcoal PKB study with or without charcoal block have many similarities in the study design, such as how subjects are recommended for the study, minimum number of inhalations, etc. And currently, the only difference actually is the administration of charcoal block. But currently, there is no standard for the charcoal dose and the timing of the charcoal administration. So that's therefore that your method for charcoal administration should be justified in your end submission. And the B is based on the 90% CI for the TR ratio for the AOC and the Cmax being between 80 to 125%. Now we think that the role of comparative characterization study provides supportive evidence for establishing B between the test and the LD products. For example, that the particle morphology can contribute to the meaning that the in vitro study, the drug performance, such as APSD and the dissolution performance. And in this figure, we see that we have 4 drug products with the same amount of the fluticasone. But the morphology, so the imaging study shows that there's different free fluticasone and the fluticasone associated with lactose. And these different association status can be detected by the dissolution study on the right. And we also, again, that the recommendation for the morphology imaging is product specific. Currently, just as I showed before, that currently this study is only recommended for the products with porous structure. The peers recommend 3 batches of T and IS products and tested at the beginning stage of the product. And the imaging in comparisons should be conducted on the deposited particles of the emitted dose. The morphological features of the particles, which may include the agglomeration and characteristics, should be evaluated. A description of sampling collection method should also be provided. So, and at the end of my talk, I prepared 2 questions for you. Question 1, which of the following statement is not true? So we have 3 options, and wait a little bit, and then review the answer. So the correct answer is C. All the studies in the alternative BE approach need to be conducted for BE establishment for most suspension-based MDIs. This statement is not true. Question 2, which of the following statement is true? We also have 3 options. And the correct answer is C. For suspension-based MDIs with limited GI absorptions, charcoal block PKB study may not be necessary. So in summary, the challenges with conducting clinical BE studies could lead to high cost and longer drug development. To address these questions, FDA has explored in vitro, in vivo, and in silico study design through the GDUFA-funded research projects to identify the alternative approaches that can be used to include the clinical BE study. And following the FDA-sponsored workshop on alternative BE approach, FDA has utilized the input received from industry and academic attendees to aid the development of several PSGs for suspension-based MDIs. And this development, the PSGs, present FDA's efforts to expand alternative BE approaches beyond just the solution-based MDIs, and highlighted the additional study consideration needed to apply to the suspension-based MDIs and also DPIs. And so I will wrap up my talk and turn to the next speaker, Dr. Ross Olander. Thank you.
Hello, it's my privilege to be speaking to you today about the in silico recommendations in our recent PSGs that Dr. Han alluded to. So I have several learning objectives for today's talk. The first is that by the end of the talk, I'd like for you to be able to identify recent product-specific guidance or PSG documents that include detailed language regarding the use of in silico modeling for orally inhaled drug products or OIDPs. The second is I'd like for you to be able to list potential roles for in silico studies within alternative bioequivalence or BE approaches for OIDPs. And finally, I'd like for you to be able to describe recent PSG recommendations for in silico study design and model credibility establishment. So as Dr. Han mentioned, there are some new PSGs that were published this year in February, and they're listed here. He spoke mostly about MDIs, but we did have new recommendations for 3 DPIs as well. And on all 5 of these PSGs, there are recommendations for the use of in silico modeling. So the first of the 2 options is an option that does not include a comparative clinical endpoint or pharmacodynamic BE study. And the PSG for formoterol fumarate glycopyrrolate is the one I'd like to draw your attention to because this has the detailed language for computational modeling. All the others just sort of refer back to this PSG. So computational modeling is not included in the 2 BE options, but there is detailed language given there to clarify purposes for why you might want to use a model. So what are the 2 potential roles that we've identified in our PSG? The first is to identify biorelevant limits for BE comparison of key recommended studies by establishing correlations between results of in vitro and or in vivo studies and in silico regional deposition predictions, and also to conduct virtual BE trials using regional deposition predictions. And I'm going to get into that in a little bit more detail later on. So if you have an in silico study that you'd like to use for your application, I've got some details here about the design. So first, a little bit about the different types of modeling you might use. You might use computational fluid dynamics or CFD modeling. And what this does is it allows for prediction of fluid and particle transport such that you can predict deposition in the different regions of the lung. It allows for consideration of realistic geometries such as a realistic mouth throat or realistic lung geometry. and can be validated with in vivo or in vitro data. So I've got a couple of animations here. The animation on the top is showing the results of the CFD simulation, and this would be the prediction of the fluid motion. And then the bottom simulation here is showing the particle motion within that flow field. And then when the particles deposit, that's when you quantify deposition in the different regions. Another type of modeling is semi-empirical regional deposition modeling. So this is essentially doing the same thing that the CFD model does. It just has a different mathematical formulation using algebraic equations and some empirical formulations. Branch-specific deposition probability is the ultimate goal of this model. And then the deposition values are summed across the branch levels to obtain regional deposition. And this is an example of what this might look like if you have the results from a study like this. One thing to mention is that this was originally developed for toxicology purposes, and some of the equations have been changed a little bit to fit for a pharmaceutical application. So how might you use regional deposition modeling to support OIDP, generic OIDP? So as I mentioned before, biorelevant BE limits. This would be testing the sensitivity of deposition in central and peripheral lung regions to differences in vitro metrics, such as APSD with realistic mouth row models or plume geometry. And the purpose of doing this would be to potentially widen the limits for in vitro comparison. If you have an in vitro study that may be failing by a little bit, you may believe that when you propagate this into the lung, that it may not constitute an actual difference at the site of action. So you could potentially use a model to widen those limits a little bit. Also, virtual BE trials, so this could be conducted almost like another study and could provide additional assurance of BE, especially if you're taking an alternative approach to what's already recommended now, but outside of one of the two recommended options. One thing I'd like to mention is that it's important when you're building a model to connect with in vitro data, and this is applicable for PBPK2. For a deposition model, the APSD results from realistic mouth throat testing or semi-empirical can be useful for semi-empirical or CFD models as inputs, but they can also be used as validation data to compare the mouth throat deposition with. If you have a PBPK model, dissolution data can be used as an input, but also could be used to assess the biorelevance of the different dissolution methods. And other relevant studies are plume geometry and spray velocity. So with these studies, these can provide model inputs as well. And lastly, it's important to ensure that your modeling assumptions and results are not in conflict with the collected in vitro data. So for example, if you have deposition data in one of the mouth throat models here, you would want to make sure that your model is matching that mouth throat model. So how can we consider variability within deposition predictions? One way would be to consider a population modeling approach. And this was an interesting paper I found from a few years ago, where they used something called functional respiratory imaging to predict regional deposition in 20 different patient-specific geometries. And this technique allows for using high-resolution computed tomography scan data to define patient-specific inhalation profiles in addition to the lung models. What you have are inhalation profiles and lungs that are paired together. And then they predicted deposition in 2 different MDIs and then compared them. So you could think of, if you're doing this to support your application, it would be almost like an in vivo study where you're selecting the proper number of subjects to power your study. Another approach could be use representative models. And we have an example here from a GDUFA-funded research project to the University of Florida. And they predicted lobar deposition using CFD models, and they had 10 different models, 2 for healthy subjects and 8 for asthmatic subjects. And they predicted using monodispersed particle injections. So these results here are showing deposition predictions in the 5 different lobes for the 4 different particle sizes. You can see the lobes are abbreviated as LUL, LLL, et cetera. And you have healthy female, HF, healthy male, HM, cluster 1, 2, 3, and 4. And each cluster was considered to be a subtype of asthma. And you can tell that depending on the particle size or the lobe, that the variability could be very little or it could be very high. So by using this approach, you could have a set of 10 models that you use to just capture the population as a whole. You're not thinking about powering your study like an in vivo study. So I'd like to say some things about PBPK modeling and how it can be used to support these products. This is a compartmental modeling approach. It allows for prediction of both local and systemic PK by simultaneously predicting dissolution, absorption through lung tissue, metabolism, and by integrating with the systemic model. You would validate this using in vivo PK data. This figure on the left shows an example of this, and in this case, there were two predicted curves, and each had a different deposition input, so you can see that deposition can affect your PK outcome. So why might we use PBPK modeling? So one way would be to understand the role of regional deposition, because while we pretty much accept that regional deposition is important for these products, it might not always be a perfect surrogate for regional absorption because you have mucociliary clearance that can move slowly dissolving or slowly permeating particles to different regions. So that you can then use this to understand the impact of dissolution and permeation, and also potentially to predict regional absorption. This also can be useful for understanding relationships between charcoal block PK data and regional drug delivery. There have been several research efforts, both within and without FDA, to understand if we can take a charcoal block PK study and say, take some of the metrics to then use it as kind of a surrogate for regional drug delivery. So you could use modeling to support that kind of approach. I'd like to say a few words about model credibility. So this is making sure that your model is doing what it's supposed to do. And in our recommendations, we have some language about the American Society of Mechanical Engineers Verification and Validation 40 standard. This was a standard developed for computational modeling of medical devices, and we've repurposed it for pharmaceutical products. So a few ideas here. The context of use is something you might have heard before. This describes what question the model is addressing and to what extent. And model risk is determined by something called decision consequence and model influence. So you can see this figure here. Decision consequence would be what's the consequence of a bad decision. So usually this is not really tied to the modeling itself. This is just like, say if we approved a bad generic, what would happen? So a high decision consequence would be for a product where it could result in death. That would be like the worst thing that could happen. Low risk would be just a minor inconvenience. But then there's also the model influence, which is how much are you using the model to actually make the decision. So if you're not using the model very much to make the decision, even if it is a high decision consequence, it might not be a high risk model because the model is only playing part of the role in that decision. And next, I'd like to say a few words about verification and validation. So model verification, this is ensuring that your numerical solutions to the relevant equations are accurate. So for most of these models, these are not analytical models. We're using some kind of complicated numerical approach to solve the equations. For example, for CFD models, this can involve comparing model predictions for simplified scenarios with analytical results. And one example is pressure drop through a pipe. So anyone who has engineering background probably is familiar with this example. Mesh and time step sensitivity, this is something sort of specific to CFD. But if you've ever seen a CFD model, this is something that's important. For PBPK models, this can look like checking the solver settings to make sure that truncation and round-off error have been minimized. So it's just essentially making sure that your model is working the way it's supposed to. But then for validation, for regional deposition, the question is, how do we validate these models and what data sources do we have? We've focused on in vivo data sources. So gamma scintigraphy is one source. This provides a two-dimensional image, and central to peripheral ratio can be quantified using this. There's also SPECT-CT and PET-CT, which can provide three-dimensional images. And this here shows an example of gamma scintigraphy where you can see the radio-labeled aerosol deposition locations. And on the bottom, there's different schemes for how you divide this into central and peripheral regions, which you can see can actually vary quite a bit from one study to another and can present another challenge. So one way we've thought about overcoming some of the limitations with comparing with in vivo studies is a lung mapping approach. And so like I'm saying, these are two-dimensional schemes, but the problem is your lung is three-dimensional, right? So if you think about that C area in the middle, you actually have peripheral lung airways that are pointing out in that area. So you need to kind of differentiate where is the actual CMP regions. And this approach that we've found from a previous study was that you can take a CT scan and then overlay it on the 2D image and kind of figure out what percentage of each branch level is falling within the CMP regions. And we think that this provides a more precise way of comparing predictions with the in vivo data. And a little word on validation of CFD modeling. We think that this should be done in all the lung regions, especially central and peripheral. The lung region should be supported by in vivo data and clinical understanding. We do have some recommendations now that give some specific ideas about how to do this. And you might need a complementary model for the small airways because CFD typically is better suited to the larger airways. So it can be helpful to pair with a semi-empirical model. And I've got some results here from a previous study that show a good comparison with central and peripheral predictive values and experimental values. I do want to note that lung mapping was not used in this case. So in this case, it still showed a good comparison, but we still believe that it can be quite useful. So I'm coming to the end here. I got a few challenge questions for you. The first challenge question, what type of in vivo nuclear imaging technique cannot quantify lung deposition in three-dimensional space? Is it SPECT-CT, PET-CT, or gamma scintigraphy? And the answer is gamma scintigraphy is only two-dimensional. The other two are three-dimensional, although there are some limitations for understanding that, and we do have a research grant going on now to help better get information from those types of studies. Challenge question 2, which of the following is not a potential role for in silico studies within alternative BE approaches for OIDPs? First is conducting virtual BE trials for regional deposition predictions. Second is to predict potential adverse events following administration of the proposed generic OIDP. And third is to identify biorelevant limits for BE comparison of key recommended studies. And the answer is B, we're not predicting safety outcomes with these type of models. That's not the purpose. So in summary, there are 5 new PSGs that include language describing the use of in silico modeling to support development and approval of generic OIDPs. And of course, as Dr. Heim mentioned, there are another 4 that were revised last month. So we have 9 now in total showing these recommendations. And in silico models might be used to establish biorelevant BE limits for recommended in vitro and/or in vivo studies, or to conduct virtual BE trials. In silico study design should consider in vitro data sources and methods for capturing population variability. And finally, model credibility should be established using a risk-based approach, as MEV-NB40 is just a suggested method that you could use another method as well. And the call to action to you is to consider using in silico modeling to support development and approval of your generic OIDP. And I have many people to acknowledge and too many people to really call out, but just want to give you a sense for how many people have contributed to this. And final thought, just on the value of generic OIDPs. This was a quote from someone, and you can see the impact that it had on their life and their family just by the reduction in cost. And next, I'd like to welcome Dr. Guo to the stage.
Okay, thanks for the previous 2 speakers, gave very detailed information. That's a lot of information to learn and also laid a solid foundation for my talk. And here I will switch gear to give you an overview of the OPQR testing and research to support GSP development. First, here's a message from Office of Pharmaceutical Quality, OPQ. Everyone deserves confidence in their next dose of medicine. Pharmaceutical quality assures the availability, safety, and efficacy of every dose. So through this, here's learning objectives of this talk. So through my presentation will inform you on FDA's own laboratory support on inhalation drug assessment. And you will understand the research efforts conducted at the FDA lab to support guidance development. And at the end of this presentation, I hope you can list several newly recommended in vitro studies as options to establish BE for inhalation products. First, okay, this topic is OPQR lab support for GSP. So who are the OPQR labs? So OPQR stands for Office of Pharmaceutical Quality Research. It's a sub-office under OPQ, and we are a lab-based office having 6 divisions. The inhalation lab and team is under Division 2, so we are located in St. Louis, Missouri. In our inhalation lab, we have a full line of equipment to do testing and research for inhalation drug products, including delivery dose uniformity testing for MDI and DPIs, Anderson cascading impactor, ACI, next generation impactor, NGI, for aerodynamic particle size distribution, APSD testing. And we have non-impaction fast imaging method instrument Sprayview to measure spray pattern and plume geometry. And we have laser diffractions, and we have various type of microscopy, including Raman microscopy, 3D X-ray microscopy, SEM, AFM, and also some other FTIR microscopy. And we have dissolution capacities and to perform dissolution, diffusion, and permeation. So what we do at OPQR Labs. So at OPQR Lab, first we support regulatory actions. So we provide product testing and a method validation program to support NDA and ANDA reviews, and also support other type of regulatory actions. And we also conduct proactive research, first to evaluate emerging technologies for characterizing inhalation product performance. And we're also involved in develop novel methods to establish better in vivo, in vitro correlations. And in the last few years, we put a lot of effort to conduct research to support product-specific guidance, PSGs, and especially focus on developing method for alternative BE approaches, including the 5 areas: realistic APSD, laser diffraction, particle morphology, and we're also exploring evaporation rate and the velocity profile for MDIs. And we've also developed clinical relevant dissolution method for MDI and DPIs. So in this talk, I will give you some examples on the first 3 areas. First is on realistic APSD, which the previous 2 speakers already talked a lot. So I will focus on more details for what you see in the lab. So traditionally, APSD was measured by cascade impactor. And the cascade impactor collect respirable aerosols with particle size less than 9 microns. So ACI is a stack of metal jets and plates to collect those particles. And between the cascade impactor and the drug product, there's a sample induction port, which shows in this picture is a metal piece with 90-degree straight bend. So it's nothing like the human anatomy. So the first area to improve to develop realistic muscle models, as the previous speaker mentioned, is to using bio-relevant muscle models, such as commercial available Alberta idealized throat, short as AIT, or models developed by Virginia Commonwealth University, VCU. And the VCU models, the 3D profile is available online. You can print it out using 3D printers using different materials. So the second area for the realistic model APSD testing is to utilize realistic breath simulation. And here the scheme is what we do in our lab to set the equipment up. Traditional way is just using a pump directly connected with a cascading impactor. So that only provides you a constant airflow. So it's not simulate the human inhalation profile. And by adding a mixing inlet and also utilize a breath simulation equipment and incorporate compressed air, we can set up simulated flow through the APSD testing. So by combining the biorelevant moisture models and the realistic breath profile, a better IVRC are expected. Here is an example for our previous study. And this study was published in 2019 on AAPS Pharmacytech. So the study is to investigate solution MDI and the suspension MDI using different type of moisture models. So our result shows, first of all, the comparison between in vitro and in vivo result are product specific. And for the solution-based MDI, all the moisture models shows close match to the clinical data. Even the USP threshold shows close to the clinical result. However, for the suspension MDI, the results are mixed. Overall, the biorelevant moisture models here, VCU models, have a better chance to give you a better IVC. So second area we spend quite a time on is to investigate the feasibility of a laser diffraction to be used as an alternative or orthogonal APSD method. Laser diffraction is a mature technique in a lot of industries. So when a particle passing through a laser beam, a larger particle will give a small diffraction angle, while a small particle will give you a larger diffraction angle. And the pros of this technique is it provides a rapid measurement. Normally, it delivers the result instantly. However, there's cons. First, laser diffraction measures volume-based PSD. Although it can be correlated to APSD, but still it's not a direct measurement. Second, is more important, laser diffraction is not able to differentiate API and excipient particles. So therefore, for complex drug formulation with multi-component, laser diffraction cannot be used as an alternative method for APSD. However, for some simple formulations such as the solution or dry powders with only API presented, laser diffraction have a very good chance to be an alternative APSD method. So there's three new products has been studied using laser diffraction. First is the Spriva Respimate. It is a soft mist inhaler. And the soft mist inhaler is a kind of product between MDI and nebulizer. The device is very similar to an MDI device. However, the formulation in the aqueous solution is similar to nebulizer. Since the formulation is a homogeneous solution, so their droplet size can be studied as a potential alternative for APSD. And our results, laser diffraction results shows a clean measurement of the aerosol with a medium particle size around 3 to 5 microns from different distance, which demonstrate the laser diffraction could be have a very good chance to be an alternative method for APSD for this drug product. The second example is a breath-actuated nasal powder on Zitra Axial. For this product, it just have a special design, and the airflow is coming from the patient mouth to blow in the air into the device. And the airflow bring the powder out, introduce the powder into one nostril of the patient, and the airflow coming out from another. So the drug formulation here is around 15 milligram of API powder only. Since the aerosol are API particles only, so it also have a good candidate for laser diffraction to be an alternative APST method. And our result demonstrated this point, and with a good clean measurement of laser diffraction, shows the medium particle size is around 30 micron. And the laser diffraction not always give you perfect result. And for the third example is a breath actuated thermally generated inhalation powder. So for this product, we had some trouble. The main point is most of the drug product was released at the first half seconds of the aerosol procedure, and it's highly condensed. The concentration is so high, and the laser diffraction measurement was not valid at that concentration. So the detail of this study has been reported in the last year's SBIA workshop, and more details were published at RDD 2024 proceeding. And the conclusion is the laser diffraction was not recommended as an alternative method, APST method for this drug product, due to the limitation of the high concentration of API particles. So the third area I want to talk about is to using MDRS, morphologically directed Raman microscopy, to characterize particle morphology and the API-specific particle size distribution. So to demonstrate Q3 sameness, the particle morphology and the API particle size distribution is critical. Here, when we're talking about particle morphology and the particle size distribution, we're talking about API, not excipient. However, the challenge are apparently for most of the product, API and excipient particles coexist in the formulation. And sometimes more than one API will be presented, and the API may have more than one polymorphic forms. And traditional techniques, such as laser diffraction we just mentioned, they cannot differentiate chemical identities of the particles. So Raman microscope is a good candidate for characterize particles, API-specified particle size and the particle morphology. However, normal Raman microscope is a very time-consuming process. And the MDRS provide a special strategy, specially designed for particle sizing characterization by implementing a morphology screening procedure before Raman conformation. So the basic idea is that morphology filters will exclude as many as excipient particles as possible before time-consuming Raman measurement. Therefore reduce the measurement time by 10 folders. At our lab, our research is focused on development, optimization, and the validate of MDRS method on different type of drug formulations, such as nasal spray suspensions, dry powder inhalers, transdermal systems, ophthalmic ointment, and the cream. And we have 2 publications in 2019 and 2021 on nasal spray suspensions. And we also have a poster on MDRS application on creams in this SBIA. Hope you'll have time to visit our poster session. In summary, inhalation drug testing and research is an integral part of OPQR's work. And our OPQR lab-based research programs provide data to support guidance development, facilitate evaluation of generic drug applications, and allow for risk-based assessment of new drugs. Here, I want to acknowledge our past and current inhalation team members and our collaborators from OPQ, OPQA, and OGD, ORS, and OGD-OB. Finally, challenge questions. First, which city is the OPQR inhalation lab located? A, Rockville, Maryland. B, Silver Spring, Maryland. C, Washington, D.C. D, St. Louis, Missouri. The answer is D, St. Louis, Missouri. Okay, that's an easy one. Second one is not difficult either. So, which of the following technique method is not considered as an alternative B approach for assessment of inhalation drugs? A, realistic APSD. B, solid state NMR. C, clinical relevant dissolution. D, particle morphology. The answer is B, solid state NMR is not an alternative B approach for inhalation. Okay, with that, I'm wrap up the presentation session. I will welcome the two speakers and the two other members to come here to host a Q&A panel session. Thank you.
Okay, we have our panel all set up. Thank you. And we welcome aboard Dr. Elizabeth Bilski, Senior Pharmacologist from Office of Research and Standards within UGD, and also Dr. Jen Zhu, Senior Staff Fellow with the Office of Bioequivalence from OGD. So we are running a few minutes late, so let's see how many questions you can get in, and also allow you a break to go do the graphic survey. Okay, any questions in-house, please? Online.
We have an online question for Dr. Langfeng Han. Does the agency suggest that it is acceptable for MDI to be non-Q1, Q2?
Yes, so as I mentioned in the talk, if it's not the Q1, Q2, we recommend that the applicants take the option 2 that follows the traditional BE approach, including the clinical study. But we also understand that it's possible that if it's not Q1, Q2 the same, the applicants can also take the option 1, the alternate B approach. The rationale for us is that to take the request that the Q1, Q2 the same in the option 1 is that we think is that the suspension-based MDIs are complex. It's likely that the non-Q1, Q2 formulation may cause the not the BE between the in vitro BE testing and the performance. So we recommended the Q1, Q2 the same in option 1 that reduce the likelihood of this possibility. But we definitely understand that it's possible that you develop non-Q1, Q2 formulation and you still see the BE between the test and the early products. But in that case, we suggest you submit the pre-end meeting request to talk with us and justify your formulation. And that's my answer.
Thank you. Next question, please.
Another question for you, Dr. Han. Are there any concerns related to standardization of the charcoal methodology irrespective of the drug product?
Yes, not a concern. That we know that currently there is no standard charcoal block that method established, and we think that you can develop your own charcoal block the method as long as that your charcoal block method can adequately block the GI absorption. And the dose of the charcoal block is not too high that can raise the safety concern. As long as that the safety and the efficacy of GI absorption blocking is satisfied, we think that the possible that the charcoal block method could be justified.
Thank you. We do have a question in-house, please.
Marie Ducharme, Learning and Confirm at Université de Montréal. Just a comment about the charcoal block. I've seen many charcoal block studies where the charcoal block will deactivate the drugs going in the lung because the charcoal block is given too close to the inhalation dose and people do not, swish and swallow enough to remove the charcoal from the oropharyngeal area. So those studies are very tricky. But you can see it right away from the PK curve because, in the PK curve, normally for those type of drugs, you see clearly two peaks. You see the peak from the lung, you see the peak from the GI tract. And when the charcoal block is not well given, the first peak is decreased. Okay?
Yes, we also see that problem that just like you say, if the charcoal block is not properly administered, it also blocks the lung inhalation. So there are some methods that can optimize. For example, that after you take the charcoal block, before the inhalation products was given, you can rinse your mouth, right? Swallow it, rinse your mouth, and that can reduce the lung absorption. Also, that you may consider use the maybe reduced amount, somehow reduce the amount of charcoal block, but I don't know if it's a timing, yes. 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 So the method, I think, definitely can be optimized. Thank you.
Thank you. Online questions, please.
We have a question for Dr. Roswalenga. What options are available to applicants to discuss issues related to in silico modeling for orally inhaled drug products?
Yeah, of course, there's the pre-end meeting pathway, which has been talked about at length in these workshops, but there's also the new MIE Model Integrated Evidence pilot program. So that offers a new way to talk about your model because you can also cover multiple drug products in those meetings. So you could have a model that you're trying to use across your whole suite of products, and we can discuss it in a more kind of holistic manner.
Thank you. And we do have a webinar on MIE on our SBIA Learn repository. Okay, next online question, please.
All right, another question for you, Dr. Roswalenga. How can applicants develop regional lung deposition models when there are no in vivo imaging data available for the drug product of interest?
Yeah, thank you for the question. This can be an issue with some products. I've talked to some length about in vivo data, and there are some products out there where you've got literature results, but in some cases you don't. So I think if you've got that situation, of course, you could try to do the study yourself, but if that's not available to you, you could try a more what we call a platform modeling approach, where you have several similar products that you do have data for, show that you have credible models for those products. And then as long as the product you're considering isn't too much different, it could still be used as a supportive model.
Thank you. Next online question, please.
This question is for Dr. Bielski. Does the agency consider alternative BE approaches for dry powder inhalers if the proposed generic product is Q1 or Q2, the same as the RLD with similar inhaler designs?
So you're asking if alternative approaches are available for DPI products that have similar formulations. We always consider alternative approaches for any of our guidances. And if you pay attention to we've been going through a lot of revisions for inhalation products. So if you've been paying attention in this arena, you'll see a lot of change going on. So I highly recommend you keep checking those periodically. And of course, if you have a DPI formulation that's Q1Q2 or non-Q1Q2, you can always come to alternative approaches. And you may see some of these option-based approaches that you see for the MDIs also being proposed for DPIs as well. I think those are, of course, always things that we will consider having meetings with you to discuss those options. And, as anything, what we're going to include for those options are all going to be product specific. So it's a really good way to come talk to us, especially if you have something you think you'd like to propose to us. We're more than happy to provide our scientific input on your proposal. Thanks.
Thank you. Next question, please.
Next online question for Dr. Zhang. Are there any differences between solution and suspension MDIs in the BE recommendations when using an alternative approach?
Thank you for the question. So for the recommendation for BE testing, I think for the solution and suspension product, they include the in vitro testing and PKB testing and CCEP or alternative study. They are similar, but from the drug product perspective, the suspension product, because it's a colored system, it's you can say it's a thermodynamically not as stable as the solution product, which is dispersed at the molecular level. So that could give you some more challenges or more studies that need to be done to do the B study. Thank you.
Thank you. Next online question, please.
Another question for Dr. Han. Will the agency accept a study where certain in vitro tests may not meet BE requirements, but the PK study shows bioequivalence?
Yes, it's possible that in the submission, we sometimes see that the applicants did not pass the solution studies, but it passed the PKB studies. And in some cases, that we will wait consider all the evidence, and sometimes we think that the PKB studies shows that the in vivo dissolution and adsorption, and in that case sometimes, but it really depends on case by case, and we accept that the submissions that fail some in vitro tests. Yeah, thanks.
I'd like just to add to that, as well as Ross pointed out, if you're failing some of your in vitro BE tests, it's a really good chance to use modeling as an option to kind of prove that it's not affecting the regional deposition or absorption. So again, even if you think you're failing and you have questions about that, please come talk to us. We'd definitely like to hear from you where your main challenges are coming from and which in vitro tests are really causing you the issues.
Thank you. Do we have more online questions?
Yes, we have a follow-up to the question regarding charcoal. Would you recommend after taking the charcoal, the subject should take some water to rinse the mouth out or not? And if water is used, will the charcoal be diluted and lost of the blockage?
Yes, actually, I think it's the good practice that you use water to rinse your mouth and swallow it before the drug administration. Of course, that you don't have to drink a lot of water to dilute the charcoal block efficiency. That is so. That means that you probably need to optimize your charcoal block method.
Okay, thank you. Next online question, please.
We have another question that came in addressed to Dr. Bielski. If Company A conducts an alternative BE approaches as per agreement with the agency, whilst Company B conducts a traditional BE studies, including a clinical endpoint study on the same generic product, will the agency consider Company B's ANDA more approvable?
Thanks for the question. I think there are multiple ways to show evidence of your local bioavailability. So I wouldn't consider one option better than the other option. I think if we have more than one option available, you can use the ones that are available. Even if we don't have those options and you have an alternative, we will also accept that. Again, there are some risks to take, right? As a company, if you're proposing something new, that might take a little bit more effort and more risk on your part because you're not sure exactly how the agency will take that. But if you discuss with us early on and you give the agency time to consider that, the more likelihood we may consider that early on. So I think it is a risk thing, right? Because if you're trying to do the clinical endpoint, we understand that's going to take time and effort and energy, but everyone knows how to do those. So again, I guess it does come down to a business decision, which way you'd like to go, but the agency considers every application as the data that it gets submitted to us. So we don't consider one more approvable than the other. It's all a matter of the evidence that you provide to us. And the more that you discuss with us, the more likelihood, you can have some success in whichever route you decide to take. Thanks.
Thank you. Another online, please.
Question for Dr. Zhenzhu. PSGs that recommend alternative study in lieu of the CCEP study, it seems that the RA-PSD has been recommended in all DAs, but other studies such as dissolution, particle morphology, charcoal block PKBE studies are selective and do not appear in all PSGs. Why is that?
Can you turn the mic on, please?
Sorry.
Thank you.
Yeah, thank you for the question. The realistic APSD is actually an advanced form of the conventional APSD that described in the USP compendium. So it's a more sensitive and reproducible and cost-effective method because it's an in vitro test than the CCEP study. And the advantage of this is it provides a more clinical relevant condition compared to the compendium APSD. And then this highlights the APSD, the realistic APSD is a critical study in the alternative study approach. So one of the important features that realistic APSD can avoid the actuation breathing coordination issue when it is tested with the actuation and the breathing has that synchronization. So so the realistic APSD, of course, it has its advantage, but also has its limitations. One of the things that we can foresee is the realistic APSD that when we test for example, the low specific resistance DPIs, it may exceed the flow rate of 100 liters per minute, which is the inertial sampling apparatus, the validation limit. So because the required peak respiratory flow rate, for a cascade impaction operation, and that's one of the reasons that the test is included in as one of the weight of evidence approach, but it's an important one. Based on like other alternative study, like the dissolution method, for example, it's also a pivotal study recommended in the PSG, but for those like the highly soluble and rapid dissolution profile drug, it may not be feasible for the dissolution testing. Yeah, thank you for the question.
Thank you. Dana, yeah.
Yeah, can I just add that? So aerosol performance for any inhalation product is applicable. So I think that's why you see realistic APSD testing in almost all the option one based approaches. It's applicable to almost everything because aerosol performance plays such a critical role, and clinical relevance is always key for us. But when it comes to other approaches, where we try to stick dissolution versus permeability, we really want to understand what's going on at the bioavailability of the site of action. If dissolution, your drug's slowly dissolving, it's going to be moving around, then the dissolution is probably the primary factor that's playing a role in that bioavailability. And that's not the case for every product. Sometimes you have something that's fastly dissolving, and your permeability is going to be the thing that's dictating its absorption properties. So that in those cases, we don't necessarily think that the dissolution test is going to tell us anything that's as relevant. So that's where those morphology characterizations fall in that place too. Sometimes morphology plays a critical role, how it becomes bioavailable. Sometimes in morphology that doesn't play as a big of a role. So I think that's where you can see the guidance getting more product specific, because we are trying to understand what's going to be the most critical product performance parameters that are going to affect that bioavailability. Thanks.
Thank you very much. And I think we've run out the clock on questions. So thank you very much for our panel. Let's plan to return on time, 2:30. Survey, graphic survey. If you parked in the hotel garage, please pick up your discount parking. You get to pay $10 for the entire day. And yeah, somebody was happy at that. So pick up your ticket at the registration desk, and let's get back here for 2:30, please. Thank you.
Test, test, test.
This should be red next.
Okay, everyone, if we could take our seats. Do a dance. Let's get started, everyone, so we could finish on time. Is this on? What you gonna do? Go ahead.
That's what they pay me for.
You know, I've always wanted to learn to whistle like that, and I never did. So I need to learn. All right, 2:30. We know it's in the afternoon session. This is the last session of the day. We have to, dedicate some time to the posters outside. That's 3:35 to 4:35. And then we have networking at 4:35. So let's learn all we could learn for the rest of the day. And we welcome our session 4 Outlook for drug-device combination products. Our speakers will be Dr. Christina Street, Senior Physician of the Office of Safety and Clinical Evaluation within Office of Generic Drugs. Then we have Dr. Jenny Wang. Oh, I don't have your picture here, that's why. Then we have Dr. Betsy Ballard, Medical Officer of the Office of Research and Standards, ORS and OGD. And then we have Dr. Jenny Wang, Visiting Associate within the Office of Research and Standards, ORS. And we will start with our first speaker, Dr. Christina Streets. Thank you.
Okay, good afternoon. So my name is Christina Streets. I'll be presenting with Betsy Ballard, the drug device combination products, methodologies for user interface evaluation. Today, our objective is 1, to provide key principles for comparative analyses and understand key comparative analyses definitions. 2, discuss FDA's experience with comparative analyses. 3, explain the human factors research being conducted by the FDA. And then we'll describe the taxonomy and how to implement it. So I'll begin. First, I'd like to start with some key definitions and information regarding generic drug device combination products. Therapeutic equivalence considers whether drug products are pharmaceutically equivalent, bioequivalent, and whether the products are expected to have the same clinical effect and safety profile for the conditions of use specified in the labeling. Therapeutic equivalence is also expected for generic drug device combination products and takes into consideration whether the end user would be able to appropriately administer the drug product without the assistance of a healthcare professional or without receiving additional training prior to use. And that's when the generic product is substituted for the RLD, the reference listed drug. This does not mean that the two products need to be identical, but the differences must be acceptable and allowable under the abbreviated new drug pathway, the ANDA pathway. Excuse me. So how do applicants and how does the FDA determine whether these differences are acceptable? The draft guidance, comparative analyses, and related comparative use human factor studies for drug device combination products, sorry about that, submitted in an ANDA, which was published back in January of 2017, provides applicants with guidance on how to conduct comparative analyses to assess differences in their proposed generic device compared to the RLD. The guidance describes 3 comparative analyses to be conducted. The labeling comparison should be a side-by-side comparison of the proposed labeling compared to the RLD, and for our purposes, focuses on the delivery device constituent part of the labeling, and that would be in the prescribing information and the instructions for use, and then any other related labeling. In the comparative task analysis, the applicant should take apart and analyze all the potential differences in the use process for the proposed generic product when it's compared to the RLD. And then the physical comparison should evaluate the physical design differences of the proposed product to the RLD, and it should include both a visual and tactile examination. To conduct a comparative analysis, it's important to first understand some of the key definitions. When we talk about the user interface, we're considering all of the components with which the end user interacts. This could include the delivery device, this includes the delivery device constituent part, as well as any controls, displays, product labeling, and packaging. The critical task is defined as a user task that when performed incorrectly or not performed at all, would or could cause harm to the patient or user, where harm includes compromised medical care. Identification of critical tasks takes into consideration the indications for the drug product, the users, such as patients, caregivers, or healthcare professionals, and the environment and other conditions that might influence the importance of a particular task. External critical design attributes are those features that directly affect how users perform a critical task that's necessary to administer the drug product. So what have we seen in terms of submitted comparative analyses? Between January 2018 and December of 2023, we received over 1,000 comparative analyses for ANDAs. The majority of the submitted comparative analyses based on the route of administration were injection products. That's followed by ophthalmic products and then oral products. We also see nasal, inhalation, and ODAC products, as well as products we've classified here as topical, vaginal, and rectal products combined. What are the possible outcomes when conducting comparative analyses? The draft guidance mentioned earlier provides detailed information regarding the 3 possible outcomes. 1, no design difference. 2, minor design difference. And 3, other design difference. No design difference is exactly what it means. There's no difference between the proposed product and the RLD. Minor difference means that there is a difference in the user interface of the proposed product compared to the RLD, but this difference is not expected to affect an external critical design attribute, which I remind you is a feature that directly affects how a user performs a critical task. And finally, other design differences. These are differences in the user interface design that may impact an external critical design attribute and therefore may impact a critical task. And the important part there is that it may impact. There we go. So what does it mean when a product's determined to have an other design difference? How does it affect approvability as an ANDA? If it's determined that the product has an other design difference, it still may be approved as an ANDA, but it may require additional evaluation. We need to consider how the design difference could impact any errors, question whether it changes the clinical effect or safety profile of the proposed product compared to the RLD. And we take all of this in consideration of the RLD as well as a specific ANDA. We look at the context of use of the product, such as the end user population, the environment of use, the urgency use, and also the frequency of use. Sorry about that. So how often do we see other design differences that are unacceptable? Of the over 1,000 comparative analyses I mentioned previously, there were 83 applications that had other design differences that were unacceptable. This is approximately 10% of comparative analyses. This slide does not show other design differences that we deemed acceptable, though. So these are ones in which the other design difference was unacceptable. The other thing that we'd like to make note here is that often throughout the review cycle, we are able to make recommendations so that these other differences are made to be minor differences or no difference potentially. And so we only send a complete response letter if we're unable to rectify the issue during that cycle. The majority of products that had other unacceptable differences were injection products, accounting for over half of the applications. Oral products made up a quarter of the applications, and then ophthalmic products and those products under the topical, vaginal, rectal were the remaining applications. When we identify these unacceptable design differences and issue a deficiency, we recommend that the applicant consider modifying the design of the user interface to minimize any differences with the RLD. If the applicant does not intend to change their design, then we recommend that the applicant submit additional data to support or justify that the other design difference will not alter the risk profile of the proposed product when it's substituted for the RLD. Examples could include in vitro studies or a comparative use human factor study. The comparative use human factor study has been an area of research interest for the FDA, and I'll pass on the presentation to Dr. Ballard to discuss further. Dr. Ballard?
So just to get an idea, a sense of the audience, show of hands for those in the audience, and I think you can vote online. How many of you have submitted and/or conducted a comparative use human factor study? So yes, you have, no, you haven't, or you have no idea what I'm talking about. So how many have submitted? 1, 2, 3. So how many have not? The vast majority, I'm assuming, have not. Okay. That's kind of what we suspected, because this seems to be a pain point when you have to consider doing a comparative use human factor study because you have an other design difference. This has been considered a pain point, and so it's one of the reasons why the research program was started. So this is just a quick timeline because I wanted to show sort of the relative the recency of this whole area of research that's the FDA is conducting. The draft, as Dr. Street said, went out in 2017. In 2020, at the start of COVID, OGD stood up what's considered the device evaluation team. And we work in the pre-ANDA space to advise industry and companies on design differences for their proposed DDCP and help them work through some of the issues and how to obtain data that will allow them to then go on for approval. So as part of that, in 2021, the requests went out for proposals to study the comparative use human factors arena. And that, while some of you in here are probably familiar with the traditional human factor studies like the validation studies or usability studies, the comparative study is different because it requires you to compare your product to a reference product rather than comparing it amongst its using your device design and having users evaluate it and make iterations and design differences based on that. So it's a little bit different. 2 grants were awarded that year. One was entitled User Interface Designs for Generic and Reference Listed Drugs, and that went to Battelle Centers, the Public Health Research and Evaluation. And the other grant that went out was for the development of a taxonomy to help identify these things, and that was granted to the University of Detroit. So the first grant, the one that went to Battelle for the user interface designs for generic and versus RLD combination products, had 2 aims. It wanted to develop enhanced methods for threshold analysis and categorization of user interface differences. And the second aim was to try and establish effective methods to assess these other design differences. Unfortunately, that grant terminated early and there are really no outcomes to report. The second grant, the development of the taxonomy from the University of Detroit, had 3 aims. The first one was to develop a body of knowledge of key stakeholder perspectives and existing strategies that people were using to assess the user interface design differences. The second aim was then to use that information and build on it and develop a visual taxonomy to assess the user interface for combination products. And finally, to use that taxonomy and actually apply it in a comparative process using a reference drug and a generic drug. So what is a taxonomy? When I took this project on, I had no idea what they were talking about. But it's a method for organizing specific concepts and creating a common vocabulary for those concepts, so that when we talk about something, everybody understands what we're saying. And the key thing that the University of Detroit collaborators wanted to do was link that design feature to a specific task and risks that are associated with that task. Okay. So what did they actually accomplish? They did accomplish aim 1. The interviews were completed with the key stakeholders. They interviewed human factors engineers, CEOs, and a variety of people, and they performed a literature search to see what was available in the public arena on comparative use human factor studies rather than the traditional human factor studies, and prepared a publication which is currently undergoing peer review. For aim 2, they did develop a taxonomy, and that's what I'm going to talk about in the subsequent slides. And they validated this taxonomy in a small inter-rater reliability study. They are developing a case report using their taxonomy and fabricated data to show how this taxonomy can be used to determine whether your design differences are other or minor. Aim 3, unfortunately, which was applying this in a broader concept, was not completed. So what is the taxonomy? So It's when they started with the combination product categories, and they used a 21 CRF 3.2E, and they used that to help define what a combination product was. And then after talking with the agency, they were able to prioritize which products they would look at in more depth. They then you looked at each of those devices and developed task analysis for each device that was very detailed. And they also looked at developing use error analysis for each of those tasks and for each of those devices. And they linked that to the URAA. They then linked that risk and the task back to a specific design feature and came up with a taxonomy. And what they started with, guys, it's not advanced. Okay. What they started with was basically look like this. And, you know, it kind of reminds you of the elemental chart in chemistry. They basically identified two major areas. One was labeling and one was user interface. They then went down and broke each of those down into certain specific categories, and then subcategories and descriptors. So the further down you get on the table, the more specific you're looking at. So on the and this is sort of a breakdown, and it probably shows a little bit better one of those particular categories and how you can trace it down. When you're looking at your device, you would start at the descriptor and work your way back on up through the thing. But they categorize each design attribute by color, size, shape, texture, and material. So very much like our guidance says, you want to look at the shape, size, and orientation of each product. And they looked at the literature and found what were the risks associated with each of those particular design features. They realized that shape and form, so a minor difference, they decided would be a change within a subcategory, whereas another difference would be a change in a critical design feature, so that it would be, the difference would be higher up or in different categories altogether. So this was obviously a very cumbersome way of doing it, and they developed a spreadsheet that incorporated each of these tasks and walks the user through a series of questions that delve further into the hierarchy and allow you to classify it. So it looks something like this. And you can see this is for a blood pressure cuff. And so you can see that they have it broken down by the group. The tasks are all there, and each one is assigned a group. And it's color coded. So it's very easy to look at, and you can just get a very quick visual as to where your differences are and where you might have to concentrate on looking at things further. They ultimately wanted to use the results of this hierarchy, this spreadsheet, and you would do one for your RLD and one for your test product, and you could quickly identify where the differences were. And then the goal of the project was then to take those differences and assign each thing a risk and come up with like sort of a relative risk value and produce what they are calling a determination report. And so in order to be a minor design difference, that it must link to a non-critical task. It cannot add any new or increased potential for harm. It cannot add or subtract or eliminate a task. It can't change the action required to complete that task. And it must fall within the same descriptor within the taxonomy. If a design feature does not fit those, then there's potential that it would be considered an other design difference. And they propose that because this could be, because each of the ultimate value of this would be the risk that would be associated with this, you could use this to help you justify why your design differences may or not be acceptable for an ANDA submission. But at this point in time, the acceptance of this report or any report such as this in an ANDA submission is a bit premature because none of this has been finalized or validated. Where do they see where do our collaborators see the future going? Well, clearly, even a spreadsheet is not the easiest to work with, and so a web-based taxonomy tool would be ideal. And they want to further take the step one step further and use those results to help you identify minor versus other. And that'll be a risk-based task-oriented assignation. They need to apply the taxonomy in a larger study. using a variety of participants, so not just human factors engineers or engineering students, but also industry reps and potentially even regulators to see in an interrelated reliability how often do we agree on the classifications. And that will allow them to continue revising and updating the taxonomy tool. So that project terminated in June of this year. And so what ongoing research does the FDA currently have in the human factor space? And what we have is an IDIQ, or an indefinite delivery, indefinite quantity. And the goal, because of all the feedback we've been getting from the agencies about trying to do these comparative use human factor studies and all the problems that are associated with them, they decided that the task one for this IDIQ would be to actually conduct a human factor study the way that our guidance suggests that we do, and following the recommendations that we give to industry, so that we could identify where exactly the problems are, what are the issues, and how can you move past them and resolve those issues, so that information can be published and be available to industry to help you do a better job conducting a human factors potential study. And the other interesting thing about this would be, once we have this data, then we could take it and look at evaluating with potentially different statistical methodologies. And that's very exciting at this point. So in summary, the taxonomy, it can be a powerful tool because it provides a common language. It can assess, the way this one was designed, it can assess the level of risk associated with the design differences, but it needs to be implemented in a larger study of the comparative process. The Office of Generic Drugs continues to fund human factors research. The IDIQ went out in March of last year. The devices are being selected, and the comparative use human factors study protocol is being developed. But what I really want to draw your attention to is the broad agency announcement that will go out in November of this year for FY 2025 to encourage anybody who might be interested in coming into this comparative use space to go to that webcast, which is, I think, November 7th, and certainly submit your proposals for consideration. And with that, we'll go on to the challenge question. Which of the following is true regarding other design differences? A comparative use human factor study is always required. The design difference may impact a critical task. They preclude approval under an abbreviated new drug application, or the applicant must modify the design difference to minimize it. And so anybody want to, do we have the results from the poll? They're not coming up, so that's what I'm asking. Okay, the answer is the design differences may impact a critical task. And challenge question number 2. Which of the following are current research projects that the FDA is conducting? The taxonomy development, the user interface design for generic versus combination products, a comparative use human factor study, or there's no current research in this space? And the answer for that is we are in the process of conducting a human factor study. And with that, I'm going to turn it over to Dr. Wang, who's going to present her topic.
Hi, good afternoon, everyone. My name is Jenny Wang. Today, the topic I'm going to talk about is approaches to analyzing comparative use human factor studies. So this is a disclaimer. So the learning objectives for my presentation today, the first one is to provide the overview of the current use of comparative use human factor studies to support other design differences. And the second objective is to discuss the analysis approach for the non-inferiority test in comparative use human factor studies. First, I will talk about the current use of human factor studies to support other design differences. So in the FDA's draft guidance, the comparative analysis and the related comparative use human factor studies for a drug device combination product submitted in an ANDA, we can see, like, in this procedure at the very beginning, during the early stage of the development, applicants are encouraged to minimize the differences from the user interface for the reference for the RLD product. And then applicants can conduct the threshold analysis, including labeling comparison, comparative task analysis, and the physical comparison of the delivery device contingent part. So the result from the threshold analysis may be no design differences or differences in design. For the differences in design, if that falls into the categories that's other design differences, applicants can either modify the user interface to minimize the differences or to provide data to support the differences, just as my colleague just presented. Applicants can provide, can conduct a comparative use human factor studies to support the differences. So for the human factor studies, it's designed to confirm that the use error rate for the critical task for the proposed generic combination product is not worse than the corresponding use error rate for the RLD. So here is a procedure of comparing the error rate of test product and the error rate of the RLD product through the non-inferiority test in human factor studies. There are mainly like 4 steps. The first step is to determine the allowable margin D by which the error rate of the test product could exceed the error rate of RLD product. The second step is to estimate the study sample size, considering assumed error rates and the margin D. The third step is to observe error rate for the critical task during the human factor experiments. And the fourth step is to perform the NI hypothesis test. So I will introduce the details of each step in the following slides. So for the first step is to determine the allowable margin D by which the error rate of the test product could exceed the error rate of RLD product. So the value of this margin D will differ between products. It's depending on the indications and the clinical consequences associated with failing to perform the critical tasks appropriately. And the acceptable margin D should be decided in consultation with FDA before the study is conducted. The second step is to calculate the study sample size, considering assumed error rates and the margin D. So in the FDA draft guidance, it provides an example using the Tangle method to calculate some power simulations given selected sample size and the alpha level equals to 0.05 and the allowable margin D equals to 12.1, just as this table shows here on the right side. So the Tangle method used in this example is the widely used method to calculate confidence intervals for the differences of 2 proportions in a paired design of clinical trials. To utilize this Tangle method to calculate the confidence intervals, usually there are several parts of information required, like the number of subjects who completed the R tasks successfully but had errors in the T tasks, and the number of subjects who completed T tasks successfully but had errors in R tasks. Also, you will need to have the total number of subjects and the confidence level. We want to note here is that the Tangle method is just one of the options for the analysis of the human factor studies. So the third step in this procedure is to observe error rates for the critical tasks during the experiment. To conduct the experiment, first of all, we will need to define the critical tasks. And then we will need to observe the error or success results of subjects for each critical task. Like on the right side, this table here, this is example data set for a given critical task, we can see like each row can represent the result for each subject. So the first column represents the results for the RLD product, and the second column represents the results for the test product. We may use like this kind of binary results, like with the 1 means like subjects complete this task successfully, and the 0 means the subject may have encountered some errors or failed this task. So after the experiment, the fourth step is to perform the non-inferiority test. To conduct this non-inferiority test, we will need to, based on the experimental data, we will need to calculate the confidence intervals for the difference of error rates between the test product and the RLD product. Then we need to compare the upper bound of this confidence interval to the allowable margin D. So if given the alpha level equals to 0.05 and the upper bound of the 95% confidence interval is less than the margin D, then the null hypothesis is rejected and the non-inferiority is demonstrated. So for the data analysis part for the comparative use human factor studies, in addition to these current recommendations in the draft guidance, FDA continues to conduct internal research to facilitate drug development and mitigate regulatory burdens for the human factor studies. For example, like our group has been working on examining like the potential alternative data analysis methods and approaches for the human factor studies. We are working on evaluating the performance of different alternative methods. So the applicants are also encouraged to propose alternative data analysis methods and the study designs for the human factor studies. So here, there are just like some examples of the alternative data analysis methods to consider. For example, non-parametric methods may be considered like the bootstrap-based methods, or Bayesian methods can also be considered to estimate the distribution of the target population's performance with the test and the RLD products. Applicants are also advised to discuss proposed alternative data analysis methods and study designs with FDA before initiating the comparative human factor studies. There are programs available for the discussions, like the Model Integrated Evidence Industry Meeting Pilot. So this is a new pilot program to offer meeting opportunities to applicants who intend to use model integrated evidence or novel data analytic approaches for BE establishment in their ANDAs. Also, the pre-ANDA program for complex generic products also appropriate program for the discussions. The ANDA applicants for complex generic drug products can request product development pre-ANDA meetings to help clarify regulatory expectations early in their product development stage. Here, I would like to acknowledge the help and the support from my colleagues and the leadership from the Office of Research and Standards of OGD. Thank you. So here is our first challenge question. So in the FDA draft guidance, comparative analysis and related comparative use human factor studies for drug device combination products submitted in ANDA, the comparative use human factor study is recommended for every application of drug device combination products. So this statement is true or false? Yeah, it's false. That's good news. So the second challenge question is, in the FDA draft guidance, so which of the following analysis methods is recommended for the non-inferiority test in comparative use of human factor studies? We have some options. First is Tango method. Second is bootstrap method. Third is Bayesian method. And the option D is the guidance doesn't recommend the analysis method for an eye test. Yeah, the correct answer is D. So Tango method was just used like in an example in the guidance. It's only one of the options for the analysis. Applicants encouraged to propose like novel analysis methods for human factor studies. And the FDA are open for discussions. Yeah, now we will go to the Q&A session. I would like to welcome Christina and our other panelists to join us on the stage. Thank you.
Thank you very much, our panelists. And we will welcome our two. Did somebody fall? No? We welcome our two additional panelists, Dr. Somesh Chattopadhyay, lead mathematical statistician with the Office of Translational Sciences. And we also welcome Dr. William Chung, who's the director of the Office of Safety and Clinical Evaluation within OGD. Okay, everyone. I'm so happy to see everybody still here at the end of the day. Welcome. Online folks, I hope you're hanging out with us as well. Any in-house questions? Acknowledged. Let's go to our online questions, please.
First question for Dr. Betsy Ballard. The taxonomy seems like a complicated way to address a complicated analysis. Does the agency think this is an improvement from a review standpoint compared to what you are seeing in submissions?
I think the purpose of trying to develop something like this is very often the determination by a review team or even an individual reviewer can be very subjective. So it might vary from team to team or from reviewer to reviewer. And if we have a more qualitative type of method to apply, the hope is that the industry will see less of that variation and have a more consistent response in our recommendations.
Thank you. Next question, please.
Great. Next question addressed to either Dr. Ballard or Dr. Streets. A risk-based assessment of human factors is very dependent on an assessor's understanding of risk. How does FDA educate its assessors regarding appropriate assessment of risk? How does this assessment differ based on the end user and context of use for the proposed product?
Yeah.
Thank you for the question. So in terms of assessment of the risk, we really look at it from a clinical standpoint. And when we look at the context of use, we're looking at who is using this product and what is the intended use for this product. So a product that is used emergently is going to have a different level of risk than one that's used chronically. And in terms of how we handle that as a division, we really try to communicate and to the best of our ability come to an understanding of what is considered a critical task and what is a potential other difference if we're identifying other difference. Our division looks at the comparative analyses, and we really try to discuss with each other. So for anything that may have where there may be some confusion about what the risk is.
Thank you. Next question, please.
We have a question for Dr. Wang. Is there a recommended allowable margin D in the FDA draft guidance?
There is not a recommended allowable margin in the guidance. So the 0.1 allowable margin was used in the example. But like for each product, I think the allowable margin is a case-by-case situation. It's related to the indications and the risk for like each critical task. So like this allowable margin should be discussed with through the consultation with FDA, and the applicants recommended to provide the justifications and the evidence for their proposed allowable margin. Yeah, thank you.
Thank you. Online question, please.
Another question for Dr. Wang. What kinds of internal research is FDA conducting to mitigate regulatory burdens for CUHF studies?
Thank you for the question. So as I mentioned a little bit, like during the presentation, our group have been working on evaluating the performance of different potential alternative data analysis methods for the human factor studies. We are evaluating like how well each method can control the type 1 error and type 2 error through simulations. Yeah, thank you.
Can I add something?
Yes, please.
So, I mean, I think, Dr. Wong's group is doing a lot of work looking at some of the just different statistical approaches. And, we heard her talk that there's lots of different ways people can approach the studies. A point that I think I wanted to emphasize from Dr. Street's talk was, we see lots of comparative analyses. We get lots of device products submitted, I think over 1,000 in the, 6 years since we implemented the draft guidance. Less than 10% of those get to the point where the design difference is something that's unacceptable. So I think that's first something to note, that really we're talking about a small set of products compared to what we're getting in OGD. And the other thing I think it's worth mentioning is we spend a lot of time today talking about the work that we're doing to support the development of the design of comparative use human factor studies, the statistical approaches and things. But I do want to emphasize, in the guidance, one thing that I think often gets missed is that, we're open to other approaches to justifying those differences. So if a comparative use human factor study sounds very burdensome, and that's something we've certainly heard from applicants, and as Dr. Ballard mentioned, that's part of why we're funding the research that we're funding. That doesn't necessarily mean that is the only way to validate and justify your other design difference. So I think that we're open to both considering alternative statistical methods, understanding the rationale for your justification of your non-inferiority margin, or if you think there's some other way you can justify why a design difference is acceptable under an ANDA and can be approvable. I think we're open to hearing those justifications and thinking about them. And I know Dr. Streets has mentioned, when we find those things in our assessment, sometimes we will ask questions, ask for more information, more justification, and potentially changes, different things. And we do want to work with you to get these products to the market so the American public has access to them.
Thank you. Next question, please.
Next question, again for either Dr. Ballard or Dr. Streets. How does a generic drug sponsor submit a comparative threshold analysis for an approved product where a PFS is proposed, but the approved RLD is a vial, or that there are no approved generics with a PFS?
Okay, so this is something that we run into frequently. And I think when you're looking at that, you just have to consider what the design features are and what their purpose is. So while it may seem impossible to do a physical comparison of a prefilled syringe versus a vial, it's really important to just provide the information that compares them, and then the labeling as well, and then also, the task analysis. It's not impossible to do. You just have to submit the information and the comparison. So I can't think off the top of my head of a side-by-side, but if there's not an applicable side-by-side component to the comparison, then you can state that.
I would just add that when we see this happen, and we've had it happen a couple of times where the RLD is a vial and they want to come in a prefilled syringe or they want to come in an auto injector. And I think in that situation, you really do need to come in early and come in on the pre-ANDA side so that you can start to get our feedback early as you continue your device development, because there may be issues that we're aware of that we can help in guiding you through that process. But I think the main thing is going to be when you have that is the task analysis, because are you eliminating tasks? Are you making it easier for the end user? So you need to keep the end user in mind and the environment of use, because if the task analysis shows that there may be some advantages, we may be able to do that. There's certainly many products that were originally approved as a vial with a syringe, with or without a prefilled syringe as part of the kit, that have now come in as prefilled syringes or as auto injectors even. So it can be done, but we strongly recommend that you come to us early in the pre-ANDA space so that we can iron out those differences.
Okay, next question, please.
Another question for either of you, Dr. Streets or Dr. Ballard. Is it possible to address differences that are considered other through label changes and IFU and not only with device UI changes?
So you cannot label your way out of a difference. But again, we're not saying that every design difference needs to be changed. So again, there's additional information that you can provide to justify that the design difference is not likely to affect the clinical safety or, the clinical effect or the safety profile of the product when it's substituted for the RLD. So the change, you do not have to make the change, but you can't just change your labeling in order to make that okay.
And there's certainly data that can be obtained and extracted from other areas. Certainly what's out in the public arena in terms of real world data for known use errors and things like that, that can be brought in to show that, to maybe justify your design difference. You can conduct your own study that may show that there's no difference, even if it's not a formal CUHF. So it's not necessarily that you have to do a CUHF if there's an other design difference and you don't really want to modify your design, or let's say you can't modify your design because of patent issues with the RLD. There are other data sources that you can use and bring in to help justify your design differences. And again, this is where it becomes very important if you're looking at something like this to come in both in the pre-ANDA space so that we can advise you early on, so that when you do get to the point of your ANDA submission, some of this has already been resolved and you know which way you have to go.
Okay, next online question, please.
This question came in addressed specifically for Dr. Ballard. If the RLD is not available in the proposed device, how does a generic drug applicant submit justification through a comparative threshold analysis?
Okay. What we usually recommend in this situation is you have to compare to the RLD, and that can be done. Certainly the labeling, you can usually get a copy of the RLD labeling, even if the product is discontinued, and you can use that to do your side-by-side labeling. You can get instructions for use and use that to do your side-by-side labeling. Where the issue becomes stickier is when you're looking at the actual device itself, because the RLD may no longer be available, but usually there will be a reference standard that can be used not only for the drug product, but also for the device issue. And you can go ahead, and we have seen submissions where they have used the physical comparison to a reference standard as additional supplemental information to support the use of their generic, because the RLD is no longer available. But we still recommend that you try and make a best effort to get the RLD product, and if not, at least use the RLD product for the labeling and the task analysis.
Yeah, I would emphasize that part, that you should be starting with the RLD as much as you can find, as much information as you can find. Photos of the RLD labeling, those sorts of things. and that some of these supportive or supplemental information from a reference standard or another generic version referencing the RLD can be part of that sort of other information that helps us evaluate and understand, is your proposed product going to be usable in the marketplace? Are people going to be able to use your drug product in place of the RLD, which, recognizing in this situation, the RLD is discontinued?
Okay, thank you. Next online question, please.
I have a question for Dr. Chung. How do you think the ongoing research work that OGD is supporting will help the development of generic combination products?
Thank you for that question. I think we've already heard a little bit about how some of this can be helpful. I think Dr. Balla's initial response to the question around the taxonomy is one that potentially can be helpful just to make sure we're all speaking the same language, acknowledging the initial questioner's framing of it is complex, but it is a starting point, and that might help get us to a place where it becomes more second nature to do these kinds of assessments, and we're using the right terminology so that everyone is understanding when we're questioning a specific design difference, we know what we're talking about. So I think that's one place. I do think that as Dr. Ballard mentioned in her presentation, designing a comparative use human factors study, I think will be very educational and informative for us to understand where are the pitfalls, where are the challenges, what are the things that we can advise on to make the assessment of these other differences easier going forward in the future. And I can't speak to the statistics. I'm not that bright, but I think alternative methodologies for analysis of data, I think, is something we can certainly look at.
Yeah, and also I'd like to add that as we mentioned, as Dr. Ballard mentioned in her speech, that there are grants, so we are working on those things also. And for the methodology, actually, I want to tell you that FDA doesn't recommend any specific method for the analysis. So anything that is appropriate for the design is acceptable. Acceptable in the sense that if it works for that particular design, then we can accept that. So actually, the word or phrase alternative method actually is a misnomer in that sense, because there's an alternative to what? We don't have a prescribed method, basically. So anything that is acceptable, that is appropriate, may be acceptable. So I just wanted to stress on that. And you can, the key is to come to FDA early, so that if you have anything, any method, any method that is not widely used, if you want to use that, or if you have any innovative ideas, you can come and we can discuss that.
Yeah, thanks for the correction on the terminology there.
Okay, thank you. Next question, please.
One more question for Dr. Wang. Could you give an example of how non-parametric methods may be used for CUHF studies?
Thank you for the question. So for the non-parametric, I would like to give an example like using the bootstrap-based methods. So bootstrap resampling may be used to through the resampling, it can be used to estimate some attributes of the target population for the statistical inferences. Then based on this method, maybe the confidence interval of the non-inferiority test can be calculated. Yeah, this is a brief example. Thank you. Yeah, so much if you want to add more.
Yeah, but whatever method, again, there may be other non-parametric methods, and but if you think of something new, just come to FDA and we can discuss that. Because Jenny just mentioned one particular one.
Next question. No more questions. All right. And let's anybody in the house has any question? They're excited about Axon? No? Okay, let's thank our panel. And okay, we do have our closing remarks, so please remain seated. And the audience, please remain seated. And those online, don't sign off yet. Okay, so for closing remarks, we're going to welcome Dr. Larry Lee, who is the Deputy Director of Operations in the Office of Pharmaceutical Quality. Dr. Lee is currently the Deputy Super Office Director for Operations and oversees research, quality surveillance, policy, quality assurance, and administrative operation functions in the Center for Drug Evaluation and Research's Office of Pharmaceutical Quality. Dr. Lee has been with the U.S. Food and Drug Administration since 2005, serving as a regulatory scientist, team lead, associate director for science, deputy office director, and deputy super office director of science. He has provided exemplary leadership in developing OPQ science, research and testing programs to support quality assessment, inspection, surveillance, and policy. So help me welcome Dr. Lee.
So I promise I will try to finish my concluding remark very quickly so you guys can get out of here, all right? But first of all, really thanks for the wonderful introduction. I do want to say that like generic, I think I was actually in Office of Generic Drug before. And I want to really emphasize that our generic is really built upon science. And the research is really a critical piece of our generic program. And our research continues to support the advancement of a generic drug product development from both a product quality perspective as well as bioequivalence perspective, because you really need both elements in order to ensure the safe and efficacy of the generic drug product. And as you probably already hear throughout today, our research support the quality assessment of various complex generic applications, also not just quality assessment, but also the bioequivalence assessment in terms of formulation approaches, manufacturing, performance, and demonstration of BE. And I want to share with you my perspective actually developing a generic, especially for the complicated one, is not an easy task, right? It's not like just like, okay, you put this and give it to patient and then look at the clinical endpoint, you get it done. No, I think it's more complicated than that. That's why our research always focus on the critical aspect as well as the innovative aspect of the drug development. This includes the analytical method for pharmaceutical, for assessing pharmaceutical equivalence and bioequivalence of complex generic drug products. Just give you an example, like developing assessment criteria for formulation and microstructure sameness of the complex generics, such as semi-solid topical products, as a demonstrate, really is not an easy task because, right, like you are trying to use those circuit to replace the clinical or the PK endpoint study, which really can help to speed up the development of generic drug in this area. And as discussed today, the agency continues to support the development of more predictive in vitro testing to establish the bioequivalence as well as the quality standard for these complex generic drug products. As I want to point out that our office actually work very closely with the OPQ, the Office of Pharmaceutical Quality, work really closely with the Office of Generic Drugs to really continue to overcome a lot of our scientific challenges in this area. And the result of the research is not just to, publish the PSG. Actually, the outcome of the research also help us to advance the policy as well as in the future regulation for the generic drug products. And then most likely, I think the industry may not realize that. And this research outcome also help to speed up and facilitate more streamlined assessment in both a bioequivalence area as well as product quality areas. So we remain committed to maintaining transparency with regard to the expectation and communication in the research area, how we actually translate those scientific outcomes to the regulatory use between the agency and the industry. And our standards for improving the quality of the submission and enabling an efficient development and review of generic drugs remain consistent. And we pretty much remain committed to really work with our industry to facilitate and accelerate the generic drug development. And thank you for all the excellent presenters as well as my colleagues for your continued contributions in advancing generic drug development. Remember, generic drugs actually about 90% of the market. So I think it's really thanks to all your hard work and really try to think outside the box, continue to make the high quality as well as equivalent generic drug available to the US patient. And thank you for your attention.
Thank you very much, Dr. Lee, for wrapping up the day. Thank you to our audience, both in-house and online. You guys have been fantastic. You would have been more fantastic if you filled out my graphic survey. but thank you all very much for being here with us. Just a couple reminders before we dismiss. Like I mentioned, the recordings are going to be posted within 10 business days. We strive for that. We may fall short a little bit depending on the workload. Your survey, which will be sent to you in the email, certificate of attendance, one pathway, healthcare CE claim code, another pathway. Certificate of attendance will support continued education credits from SOCRA, RAPS, SQA, and ACRP. You'll get, all attendees will get one email within 24 hours. Please allow us 24 hours before you start sending email inquiries, because we'll still be here working on the second day. So 24 hours, you'll get one email. It will have the survey link for the certificate of attendance. It will have the link to get into the CE portal and also the claim code for day one. Every day, we'll send a different claim code for healthcare professional attendees for the CE credits. And everything, survey, certificate of attendance, CE claim code, everything has a 2-week deadline date on it, October 9th. As soon as you get that email, please complete it. And that's it. Well, it's actually October 9th. So that's it for in-house, for online attendees. Thank you very much for being with us. We can end the broadcast for online attendees. For the in-house